I will say, hello, interwebs. It is I, Hewlett, David Hewlett, back with another I'm with Stargate Genius. And today we've got Bas de Mooij. No, I said it wrong. How is it?
De Mooij.
De Mooij.
Bas de Mooij. We're close enough. So
I even asked in advance and then screwed it up anyways. But
don't worry.
Bas, welcome. First off, thank you so much for doing this. I am really excited to get into, because you're down where the sausages are made, basically.
Yeah, pretty much. Yeah, we're on the floor.
On the floor, exactly.
We do the dirty stuff.
Exactly. All this highfalutin, concept stuff. You're actually doing the work.
We do the actual science.
Let us know who you are and what you do, and then we'll dive in.
Fair enough. My name is Bas de Mooij, and I work as a research technician in the lab of Franck Stahl, who was a previous guest on the show.
Yeah, fantastic chat.
And it boils down to he has the idea of what we want to investigate. He talks to us about it, and then we perform the experiments. So I do all the pipetting in the lab, make sure all the cells are ready for the experiments, and actually, perform everything.
You make that you sound very diplomatic about that, because the reality is he's like, oh, let's do this, and you're like, oh God, how do we make this happen?
Yeah, he has some, and I think most professors have that, but they have the grandest ideas, and then he talks to us and his PhD students about it. Okay, guys, I have this idea. I want to investigate this. How are we going to do it? And then together we think up a plan of like, oh, we can start in that way, or we just straight up tell him that's not going to work, or that's impossible.
You're the dream killer. You're the Franck Stahl dream killer.
Yeah, it's like, no disrespect to anyone, but usually the professors haven't been in a lab performing experiments for years upon years upon years.
Interesting.
And they sometimes lose sight of how difficult things can be.
Right.
Which is not a bad thing, but yeah, it does take some reminding occasionally.
Well, and if you're not in it every day, it's a bit like a language, isn't it? You sort of lose track of the nuances of it and stuff. I can totally see that. But you love it. You love the lab.
I do. Yeah, I think it's the best part of science.
Yeah. You tried to leave the lab, and then you got dragged back.
I actually did, but that was in between lab works. When I graduated from university, I had the idea of, maybe I want to do a PhD, get further into research, and then it was a difficult time, so there weren't many positions. And at some point, while applying to position, I go, the follow-up I have to do is, the further along I get in my career, the less I'll be spending time in the lab and more giving presentations, writing grants, writing papers. It's like,
oh God,
no. Yeah, oh God was probably the best reaction to that.
I don't think my wife does a lot of the paperwork stuff, and I don't know how she does it, because I just don't. It drives me mad.
Some people really like it, and that's good. So it leaves me the space to do the lab work. So
we get to play. It's perfect.
Yeah,
you obviously really love it, don't you? I mean, it's something you're quite.
Yeah, I've been doing it for, let's say, almost 10 years now, and
really,
I wouldn't change it. I don't think so. I tried in between. I tried teaching a little bit, but after half a year, I was like, I missed the lab. I want to do it myself.
And we'll have to talk about that, because that's an interesting distinction. I mean, I think there's because you obviously do. I mean, even just in our pre-chat, you obviously like explaining this stuff. You like talking about this with people. So I could see the lure of education, but it is a very different. It's not just that, is it? Every job's got all this stuff around it that makes it more.
Yeah, no, absolutely. It was, and most for the teaching, it was divided into two parts. We had the theoretical lectures, and then the practical stuff where you teach the students the lab work.
Oh,
that part was amazing.
I was gonna say you loved that part.
Yeah, that was good. And I still enjoy when we have students over, explaining them how essays work, etc.
I'm coming. I don't know what you're going to infect me with, but maybe a whooping cough.
We'll figure something out. I'll tell you, you won't be the oldest student we had.
Oh, really?
Or I've ever had. Yeah, recently graduated one. I think he turned 61 during his internship. Assuming you're not above 60.
No. I just look at. Yeah. That's interesting, because it's always one of those things that I wonder if I should be considering going back. But this just, to me, in a weird way, I feel like there's too much to learn. I don't have time to go back to school for one thing.
A lot of people I talk to, when I tell them about what I do, they're like, I could never do that. But I always say, and a few colleagues of mine as well, if you're good in the kitchen and you can follow a recipe step by step, then you can work in the lab as well.
I'm out.
Okay, never mind then.
I can burn water. I'm like, it'll be interesting. I'm really looking forward to visiting because I do love, I've been playing with some of these little biology kits and stuff, little pipettes, and played with that. I love that. I love, again, the hands-on stuff. I think there's something about understanding the nitty-gritty of this stuff that I think helps you understand what's going on better. Then you sort of, I have much more appreciation for all of the robotics now that allow you to do this stuff on larger scales. Let's go back for a second, because I want to, I kind of want to know how you got into this. I want to know what sort of instigated it. Were you mixing up little experiments, chemistry experiments and things as a kid? Where did this come from?
No, not really. I think it started with my mother always made me watch Voyager with her, Star Trek Voyager. That's what got me into science fiction.
You were brainwashed.
Yeah, she really loved that show, and I was just watching. She never forced me. I'm lying with that, but I started watching that as well, and then gradually I migrated to Stargate. Of course, I have to mention it.
Wow. Okay. Great.
Yeah.
SG-1. You're watching SG-1?
I started with SG-1, and then, of course, when Atlantis released, I started watching that religiously as well, through some very creative means, I'll have to say, because it never aired here.
Oh my God, it's so frustrating. I know the release stuff is insane. So were you drawn to the medical science aspect of those things, or?
Yeah, a little bit, and just the science in general of discovering stuff. And during my high school years as well, I was more interested in biology and chemistry compared to the economics or geography and stuff. So that naturally, veered me towards the science profile in high school. Okay. And then at some point, the director of the Leiden Institute for Chemistry at that point, he gave a lecture in our high school about a new study they were going to set up called Life Science and Technology, and I was like, yep, that's for me.
Wow.
And that's how I got into it.
It just sold you hook, line, and sinker right there.
Yeah.
God, it's so important. The outreach stuff is so important, isn't it? And they've had you since, basically. Like you've been
pretty much. Yeah.
Now, were you there because originally was it not specific to immunology? Was it originally, and then it became the medical school, or was it always just part of the medical school?
No. The Leiden Institute of Chemistry was one of the faculties where I did my education, and that was more focused on microbiology, molecular biology, and a little bit of cellular biology. And then after I graduated from that, during that I did some internships, and after I graduated, I applied for a job at immunology department at the LMC. And there I got the position for a technician.
And what is it? Okay, so what does that mean? Like, you are a technician? I don't even know how that translates into a job.
A technician is it's pretty much a job title for the people who perform the experiments.
Okay, so if you're hands-on, you've got the technician role, basically. Yeah. Okay,
and then you work with a lot of the PhD students, and they are going for their degree, and they do a lot of lab work as well, but they have more theoretical background usually. And
you're helping them out with that stuff?
Yeah.
Oh, great. Okay.
Yeah. They, together with the postdocs, so the people who already did their PhD and hold a doctorate's degree, in combination with the professor, the postdocs, and PhD, we think of the experiments, and then usually the PhDers together with technicians perform the experiments.
Right. So you get to be involved in basically all the stuff that's happening, or do you have specific areas that you each work in?
In our group, we're quite a big group, so I have my specific fields of expertise. I don't know all of the techniques that are going on, but I at least am involved in a lot of the discussions around it.
So how many people are we talking about? How many are there?
I think Franck Stahl's group is about 25-ish
people.
Wow. Maybe a bit less.
And is it like one big lab, or is it split up into lots of little ones, or what's the?
No, I think we have one big lab and then two smaller labs which are more aimed at specific types of work. So we have one ML2 lab, we call it, and that's I don't know if you ever heard of the biosafety levels.
No.
It's biosafety level is an indication about what kind of material you can work with.
And like how dangerous they are.
Yeah. Ebola virus is like I think 3 or 4. Then you have Marburg.
Really?
All the nasty ones are very high up.
Yeah.
And Emil, you have to be in a suit. It has to be.
So what are the levels? How do the levels work? So like Ebola is like, I guess, top of the list.
I don't know all the details about it, but the basic one is no safety. So that's very basic material, non-infectious, anything. Then you have levels 1 through 4 escalating about to infectiousness, danger to the environment, all that kinds of degrees.
And where are you at? What's yours?
I mostly work in the 1 and the 2.
Okay, all right.
2 is the highest we work with.
And what does that mean for you?
A lot of viral material. ML2 is very lenient. It's a bit more strict compared to 1 with safety levels. You have to work in a laminar flow hood. You have to be a bit more careful, disinfect your material in a different way.
Right.
And then from 3 and onwards, it starts getting really complicated with actual airlocks and stuff.
That had no interest to you? You're not interested in going beyond that, or your personal safety is?
We do have them in the LUMC, but I've never had the chance or applied for a job that I could work with that.
Right.
And it takes some specialized equipment and people to do that too.
God, it just seems like it'd be so uncomfortable being in those suits. So I remember just being in a hazmat suit for a few minutes at a time on Stargate. It was just became miserable.
Yeah, I can imagine. I've never done that, but a couple of my colleagues also work in a clean room, which is different, but you can imagine you have to suit up. Everything has to be as clean as possible, and that's where they make the patient products.
Yes, of course.
The actual medicine or treatment that goes back to the patient, so it has to be as
the actual gene therapy would be built there. Is that
well, we do the basic research, of course, and in the ML2s, etc. But once you start making the actual product, you move it to a clean room, and from there on, you work with it.
So, what's a day for you? Like, what is it? What is it like? Like you go to work, you grab a coffee.
Yeah.
I'm assuming. I don't know.
Well, tea, but I don't.
Oh, you're a tea.
Yeah, but similar.
Was it like an English tea? Are you an English tea? What do you? Was this like
as strong as possible? I really like Yorkshire tea, for instance. But you can make Yorkshire tea. Yeah,
that's my tea. That's the one.
Yeah, it's amazing.
I love it. It's the best. Yeah. It's like a cup of coffee, only it's tea.
Yeah. I drink it from a large Guinness mug, so I just pretend it's Guinness.
Well, look, I have a ZPM of coffee. That's how I work. Yeah. Oh, nice. Hey, it's the Guinness. Yeah. Are you a Guinness fan as well? Do you like your Guinness?
Yeah, I love it.
My son's almost like a fan, if that makes sense. Like it's more than just drinking it now. It's like it's we'll have to partake together when we.
Sounds good. There's a good pub in Leiden, and a couple of them have it from the tap. So.
Well, and we have, and you also know how to pour it there. In North America, for some reason, it's very hard to find a well-poured Guinness.
I think these are it's an actual Irish pub, so
that'll do it.
Yeah. If you can't pour a Guinness, you're probably not allowed to label yourself an Irish pub.
Exactly. So, you get yourself in a lot of trouble if pouring that wrong. It's so. Genius beers aside, we'll do that. We'll definitely do that when I'm over there. But so how does the day work then? So you've had your tea.
Yeah. Well, I come into work. I check my agenda, emails. What do I have to do today? If it's something simple, I occasionally forget. I talk to I keep track of everything I have to do in a day in my agenda, because you know how that goes, right?
Now, are you a paper agenda guy or are you a digital agenda?
No, I have everything in Outlook. Microsoft Outlook is what we use. Yeah, I don't prefer it either, but hey, that's what I get from the job.
Yeah.
Okay. So I do that. I check my emails. I answer the urgent ones, and then I go about my day. Go past colleagues, what they have to do. Can I help with someone if I have time?
What would come in on an email? Like, what are you looking for? Like, what is like? I mean, when you say you've got to put out fires and stuff,
it's different. It's people looking for reagents that they have too little of, and I help them out if we have any spares. It's colleagues. Or with urgent requests, can you help me with this experiment? I'm a bit short on time. Can you take something over?
Oh my God! And if they're students, it's all going to be last minute too, isn't it?
Mm-hmm. Some students, yes. Some students are very good.
Yeah, I'm sure.
It helps that I'm usually not the one who is a daily supervisor as the students. It's
right.
They're mostly assigned to the PhD students. So
right. Okay. So you deal with students a lot in general, though. I mean,
We usually have 1 or 2 walking around. Yeah.
Yeah. Okay.
We got some new ones coming next week. So.
Oh, really? Do they have
looking forward to that?
Do you haze them or anything? Is there any kind of like?
No.
Oh, okay.
No. Usually we don't have any say about the matter. The professor just picks them out.
Right. That's great.
But so can't complain so far.
Well, and that's got to be nice too, because you've got sort of like fresh perspectives and fresh eyes on stuff as well. It's got to be kind of fun,
absolutely.
Keep you on your toes.
Some students are really good with the questions, and they're like, "Oh, why don't you do this?" I never thought about that. So that definitely helps.
It's funny. I talked to I talked to a bunch of students at a science fair in Canada. There was this Canada-wide science fair, and almost without an exception, I asked how they, I was like, "Oh my God, you're working on cancer cures. You're doing this. You're doing that." Mm-hmm. These are like, 15-year-old, 14, 15-year-old kids, and they all said, I went online, I looked at a bunch of YouTube videos, and then I emailed the guy or the woman who was doing the work, and that's how I got into it. I feel like it's all there. If you're interested, you can. You can learn a lot of this stuff. You just need to get people excited enough.
Definitely. What I always do is a lot of people are interested in reading like the scientific papers, etc.
Yeah, I try.
But then they get hung up on the fact that they usually have to pay to get access to it.
That's criminal. Don't even start me on that. That seems so wrong to me.
What is a very good workaround is that you just email the authors and like, "Hey, I want to read your paper. Can you send it to me?" And usually they're very happy to do that.
Really?
Yeah, absolutely.
Because I always feel like I'm being such a pain in the ass if I'm doing that. Like
most scientists I know are like, "Oh, someone is emailing me to read my paper. That's awesome."
Do you email them directly, or you go through whatever the?
Usually, if you look up on like PubMed or whatever, there's a list of corresponding authors,
and just email
them. Wow. Like, "Hey, I want to read your paper, but I don't have access. Can you send it to me?"
Oh, that's fantastic. Yeah, I love it.
Otherwise, email me and I'll send them to you. That's also fine.
Well, it's so funny because I have my friend who's also a professor is the same. He says, "Oh, just email me. I'll just James Gurney just says, but it's a very odd system. That okay. So going back to your day, you're coming in, you're answering these questions, and then do you have a set time in the lab or
no, not really. It really depends on what I have to do on the day. Sometimes we have meetings, like work discussions with the entire group.
Right.
On Monday we have that first thing in the morning. We sit together with everyone, see if anyone has an announcement.
Do you dread it or do you love it?
It depends.
Yeah.
Sometimes it's fun. So if I'm busy, I dread it.
Right. Because it's taking time away from what you need to be doing, right?
Pretty much, yes.
Right.
And but sometimes there's someone in the group I don't work with as much. They have a presentation. They tell us about what they've been doing for work, the problems they run into, and together we can have a discussion on how we can alleviate those problems.
Oh, neat.
Continue research. Yeah. So we have that, and then
people are open to that. Like they present this stuff, and
that's part of the job. Because you can't advance science if you don't work together.
I know, it's so funny because Hollywood's got it so wrong on that. I mean, like McKay is a perfect example where it's always like one guy solves everything, and it's like, no, it doesn't work like that.
Don't get me started.
Yeah, I know I see it now, and I think it's quite damaging too, because I feel like it's also a very, again, I would argue this is a very North American thing as well, where it's the onus is on that brilliant spark, and if you're the person who can come up with a brilliant spark, but the reality is it's all the work to make it happen is actually
of course you still need the idea, but what I was going to say, that science takes a long time to properly show on screen anyway.
Yeah, that's the other problem, is we're too impatient. We don't want to see, it's a montage and then you've cured cancer, basically. Not the way it generally works.
No. And the news doesn't help with how do you call it, making stuff seem way more exciting or progressive than it actually is.
Yeah, it's the nature, unfortunately, of sort of the entertainment news, basically, is that everything has to be like, they've cured cancer. Then you read about it. It's like, well, they've made a breakthrough in something, perhaps, but they haven't, this isn't, it's like we found another Earth. That's the other one I love. It's the same. in the same way that the biotech stuff is always about curing cancer, or making people live longer, and the space stuff is always about, we found another Earth, and it's like,
it possibly harbors life.
Yes, exactly.
Yeah, or it's in the Goldilocks zone, but it still doesn't say anything.
The number of times I've heard those announcements is just is crazy.
Also, a lot of people forget they're not rodents.
Right. Yes, that's true.
A lot of potential cancer cure, and then you read the paper or the news article, and they did it in mice, and it's like, okay, how's that going to translate to humans?
Yeah, it is weird, isn't it? Because that is something. There's this. Again, I think there's a connection there probably because of the way the information is presented, that we believe that if you can cure it in mice, you can cure it in anything.
Yes.
Which is obviously not the case.
Yeah, you got to start somewhere. So
I love that. Though. I feel like you should have a T-shirt that says "Not a mouse." You know what I mean? Like it's like
and as much as I hate mouse experiments, we're always going to need a certain level of it. Sadly, you need to start somewhere. You can't just instantly start testing it on humans.
Right. In fact, there's been some recent issues with that. There was a couple of things. There were a couple of trials in China, I believe, that went wrong, and there's this whole debate as to whether should you publish failures as well as successes.
I feel you should, and
me too.
That's one big gripe. Like, then again, I wouldn't call it failures per se, because negative data is still data. It's just the way we did it this way hasn't gotten the outcome we wanted, but you should still be able to publish it so people can see, they tried it already, doesn't work. We're not going to do that anymore. Now you run the risk of people, we're going to repeat it and then run into the same issue, working.
That's exactly
and then waste a lot of time and effort.
Yeah, and potentially make some people very sick in the process that have already been made sick in the process.
Yeah.
I mean, it's and again, that wasn't to point the finger at China. I think it was just we are struggling with this idea of how do we. There's an impatience. People want to get this stuff,
which obviously. I get, because of course you want to get it done.
Yeah, especially when you're dealing with a childhood disease that can be. I mean, the stuff that you guys are working on is extraordinary. All right, go back to your day, because I'm very good at keeping you off topic. So
that's
right
fine.
away,
I do the same.
I've destroyed your day already. You thank God I'm not there, because if I was there, you get nothing done.
The only thing is I'm not able to cook for my wife today, so I'll figure that out later.
Ooh, man, I feel bad taking someone's dinner away from them. That is not, so she's in charge of dinner?
Yeah, but she's probably reheating something, so I wouldn't worry. She's well taken care of.
Okay, all right, good. As long as she's not hungry. You don't want
no.
Hungry spouse is never a good sign.
They say happy wife, happy life. So.
That's it. That's it. And for me, it's just feed me. That's
very good.
So okay. So when do you get to the lab? When do you get to what time of the day is this?
The regular hours are usually between 9 and 5.
Right.
But it's very flexible.
I was going to say, because science doesn't work 9 to 5, really, does it?
No, absolutely not. If you have to come into the weekend, you have to come into the weekend. You can plan around it, but sometimes the cells, etc., are very fickle.
Yeah, sometimes. Like all the time, aren't they?
Sometimes. It depends on where you're working with, but in general.
Right.
9 to 5 is regular day. Everyone tries to be here in around 9. Some people come in a bit later and then stay later, of course. But if you have to work longer, let's say you work from 9 till 8 or 9 because your experiment has to be finished, or some machines are only available in the late hours of the day.
You can always my favorite one, that cytoflow. What is it called? The flow. God, what is
flow cytometry.
Oh, yeah, brilliant. That thing is
lovely.
I watch videos on how it works because I'm so enamored.
Can you explain it to me how it works? See if you understand it.
Well, if I've got this right, you take your sample, you mix it with some kind of an agent that allows it to go through to be sort of injected into the machine in such a way that it reduces it to like basically a cell. You fire a laser at it. The laser, then detectors will pick up either the backscatter and the side scatter. And from that, you can determine how large or complex a cell is and identify it like that. But you can also, apparently, this is the one I didn't know, apparently you can also add fluorescent to it. And then some of these ones will actually pick out numerous different types of tagged fluorescent elements as well.
That's pretty much it.
Is that it?
Yeah.
I mean, who thinks of that? I know. Right? Let's drop one cell at a time Am I saying that right? Is it one cell or is it one particle or what is it?
No, it's one particle, but the cell is in this case a particle.
Okay,
And
all right. I don't know how they do that even. Like, how the hell do you get?
Yeah, they focus it with a fluid next to it, and then it goes into one by one. It is insane how it works. But the machines we have now, I think they can register about 10 to 15,000 cells a second.
Which is insane.
It's insanely fast.
You got to be excited about that, though. Like, I'd be playing.
Oh yeah, I love working with those machines.
I'd literally be playing with that.
And there's even a better version of it in the labs we have, and they can sort the cells.
Oh, so they just knock them into different things as they.
Yeah, that's pretty much it. You have the one by one. They go through it, and then the computer detects, oh, this cell is, let's say, a T cell or a B cell.
Yeah.
And then later on, they get magnetized, and they go either to the right or to the left, so you can sort them in the specific tube. So you have only the T cells in your sample or the B cells, and then further on, you can work with that further again.
Oh my god!
It is ridiculously complicated, but it's really fun.
It's ridiculously complicated and yet strangely simple as a concept. I mean, like, I love the idea that you're. I mean, now, but hitting them with lasers doesn't bother them. It doesn't.
No. It's well, it's not ideal. Of course, you're still pushing them through a machine.
Yeah.
So they won't be as happy as they are comfortably in their growth medium. Sure.
Right.
But
some of them might get damaged in the process.
Yeah, but in general, the lasers don't harm them.
Okay.
Because they're of a specific wavelength that usually only the fluorochromes that are attached to the cell can absorb them.
Oh, okay.
And then they get excited by it and then emit a different kind of frequency, and that's how we detect them.
That's the ones you tag. Those the tagged ones you talk about, right?
Yeah. Let's say you take a T cell. It has.
All right. Explain to people what a T cell is. That's the killer T cell. Are we talking? Is that?
That's that's a subtype of T cells.
Oh, is it? Okay. All right. Okay.
Yeah. You have in your blood, you have pretty much 3 components. You have your red blood cells, your white blood cells, and your plasma.
Right.
The T cells are part of the white blood cell compartment, which is the immune system you have in your body.
Yeah.
And there's different kinds. You have T cells, B cells. Those are usually the types that people know. NK cells, monocytes, macrophages. You can there are numerous of them, but the T cells and the B cells are usually the most well known.
Okay, they're the ones that will come to a problem and deal with it.
Yeah, usually the other cells actually are there quicker, and then they signal molecules where T and B cells get attracted to it. It's a whole difficult system. Yeah.
How does it work that fast? It's something I've never quite understood. How does it? How on earth can you?
Yeah, the most of the the macrophages, monocytes are already primed to detect these incursions, I'll call them. Every cell type is focused on a different kind of infection. So you have bacterial focused, viral, fungus, etc. It's really complicated. I don't know all the details about it.
But you know more than most because you've studied the process of like a whooping cough forming and stuff, right?
That's true. Yeah, that is my first job. I worked on a project where we investigated the effects of the whooping cough vaccine, so pertussis, on the reactions of the immune system.
But this sounds like mad science to me because it's like, hey, come on in, we're going to infect you with whooping cough. I'm like, who signs up for that? Obviously not a hypochondriac like me.
you get a generous payout for it.
Oh, do you?
No, you do. No, I'm not. I don't know if it's generous, but you do get a volunteer.
I got through college by getting infected by various different things.
It's not as bad as in America where you get money for donating blood.
Right.
But you do get a compensation of a little bit for usually taking part in those studies, and then it ranges on how high it is based on how invasive it is.
And I should say, I'm sort of making fun of it, but the reality is it was in a very safe environment. There was antibiotics standing by. You're ready to go.
Yeah.
But basically, your first job was infecting people with whooping cough, and then
I didn't do the infection myself. That was done in England.
Oh, was it? Oh, okay.
Yeah,
okay.
And then they got the patients there, or the how do you call that? The volunteers.
Right.
I was going to say victims, but that's not the proper term for it.
But sure. Okay. Yeah.
No, the volunteers, they signed up to join the study, and then they got infected with different doses of pertussis bacteria. And at certain points, they were all in-house, so they were kept in the hospital for 2 weeks, I think.
Right. Get a lot of reading done.
Absolutely.
Yeah.
So that's one benefit of it.
Yeah.
At certain time points, they draw their blood, shipped it to us in the Netherlands, and then we processed it to analyze the white blood cell compartment. So the levels of T cells, the levels of
Using
B
your
cells,
cyto,
etc.
using your flow.
Yep.
Sweet.
Yeah.
Yeah.
We have a number of panels, we call them. That's let's say see it as a different collection of antibodies. They bind to the enzymes or the proteins that are on the cell surface, and each different type of immune cell has a different collection of these proteins on the surface.
And you're watching every stage of the infection process.
Yeah, a couple of days. So I think we had day 0, day 1, day 3, 7, 14, and 28 probably.
See, right away, I would never sign up for anything that starts with day 0.
I'm probably, I get that. Yeah. I'm probably missing the details, but it is all in the paper, and it's been years ago, so I don't know the details.
Yeah, I tried to get, I started reading I try to read the abstracts at least, and then I get AI to help me with the rest of it. I found even the abstract on that one very difficult because there's a lot of, as always with biology, there's a lot of terminology.
Yeah, it's very complicated, especially if you're not in the field, and it's very difficult sometimes.
But the basic premise was you were trying to figure out why this virus.
Yes, it's bacteria, I think.
Is it bacteria? Okay.
Yeah.
Why some people were spreading it even once they were vaccinated, and other people weren't.
Yeah.
And you found that it was a difference in the way that the body first reacted to it, or something.
I think that's a combination of it. It's the same with COVID, which of course is a virus. But what we did in that pertussis study, we were monitoring like all the blood samples we took, and then we could clearly see on day 0, very low response for some cells, very high for others, and then you can graph out all the cell populations in time.
That is so cool.
Yeah, some of them came up after 7 days, some of them at 4, some started at day 1 very high, and then wavered off in the long term. And it's really
cool. You're just
looking
at this as you're putting this stuff in the machine. And you're seeing these graphs for these patterns of recognized cells showing up. And where were you at in your career at this point? Where was?
When I was working there, that was during I think one of my first few years of working as a technician.
Wow.
So starting out as a newbie. Really.
Did you learn how to use the machine for that?
I used it very sparsely during one of my bachelor internships, which was about 6 or 7 years. 6 or 7 years prior to when I started that job. So I had to learn a lot of it, and especially the more
hell of a manual
stuff. Yeah. I had good colleagues teaching me everything, so that really helps. That helps. Yeah.
Are these machines becoming? They must be becoming more intuitive as they get more advanced. Is that, or are they still pretty?
They're still very complex, and of course, it also depends on how complex the machine itself is. But you can work with the software. Of course, every machine from different supplier has a different type of software.
I can imagine that must drive you nuts.
And some are much more intuitive than, especially the newer machines are very, very, very easy to use in comparison to the older ones.
Do you have a favorite?
Currently the Auroras.
Yeah.
They're from SciTech Bioscience.
And they're the ones that can knock the things into the right.
They have a machine to do that as well. We're testing.
That's like an add-on. It's like modular. You can add these things.
No, they're usually different machines because they work a little bit differently internally. So you have to have both machines. Although you can do the analysis on the sorters, but not the other way around. You can't sort on the analyzers.
Right. That seems like a whole other system, doesn't it?
Yeah.
God, it is amazing. Like the leaps and bounds we've made in this, it's just extraordinary. Okay. Once again, I've taken you completely off your day.
It seems to be the theme.
Yeah, that's how we work. Okay. So you're not in the lab every day then?
No, I try to be.
Yeah,
but it's not always that a day requires you to do experiments in the lab. What we do then, we spend our time analyzing data, updating protocols.
It's a lot of data too, because the better these machines are at getting stuff for you, the more information is going to be to plow through.
Exactly. Yeah, and it takes a lot. We have a couple of people in our group that do solely bioinformatics work.
Yeah.
So from
I've got
next
a friend
generation,
who used to run a company that did bioinformatics. Amazing stuff.
It is basically big data.
Yeah.
It is really big data.
Do you have any interest in that at all?
Not really. No.
The results. You like the results, just not.
I like the results, but not the processing. I'm also not very good at it, so that doesn't help.
It's for some reason the camera seems to have decided that it's drifted off. Or it might be it's light. It might be the light. It's looking for light.
It could be. Let me see if that doesn't. Well, it helps a little bit.
See, there you go.
Yeah,
I've been there. Now you look cool too because you got like a half. Yeah. Okay, so that's interesting. So you're not in the lab. It's funny when you say technician, I would have thought that you just get right in there. You're in the lab right away. But of course, when you're dealing with this kind of data and stuff,
it does take a lot of prep work as well. So we have to write our protocols, think of all the samples we need, the proper controls with it, which is very important, and a lot of people tend to forget.
Right. Oh, really?
Yeah. If you don't have any controls, you can't say anything about your data.
Oh God! And people will literally forget to do that.
Yeah, but usually on accident. They don't.
Right. Okay.
Intentionally do that, but it does happen. And usually, if you forget them, you can still manage to at least tell something about it. But you usually can't publish it in a journal if you don't have any proper controls.
Are you meticulous? I mean, has this made you meticulous, or are you meticulous already in this stuff?
In general, I am.
Yeah.
But I do, as you get more experience, you know where to cut corners or where you can cut corners.
Right.
Which is not always
that's
helpful.
more optimizing than being.
It's probably a better way to put it. Yes.
Yeah.
It does make it difficult when you get a new person and they're like, oh, but it says this in the protocol. Why aren't you doing this and this and that? And it's like, yeah, how do I explain that?
It's a waste of time.
Yeah, pretty much.
No, but interesting though. Yeah.
The first time you do a protocol, always follow it to the letter, and then you can adjust. Same with cooking a recipe, like I said.
Yeah,
that's around the kitchen.
That's true. The first couple times you do a recipe, you tend to go exactly by whatever the New York Times is telling you, or whatever. And then once you've got a sense of it, you get a feel for it, don't you? And that's the same thing. Interesting. That's the same thing.
Then you can modify it. Yeah, because then you know how everything works and blends together.
Yeah.
And then you can add to it or remove some stuff.
I could see why you want to get in the lab all the time, because it's got to be, there's a certain sort of hands-on element there that's just got to be,
It's the most fun part.
And what is it you like about it? Like, what is it about it that, what is it that got you?
That's always difficult, but I think it's just the job itself, as in the things you perform, the handling, and then actually seeing the results come out of something you do.
Right.
I think that's also partly why I don't like the bioinformatics too much. It's just you enter your stuff in the computer and then something rolls out. It's still very important, don't get me wrong, and I'm happy that other people seem to love it, but it's not for me. I actually see the cells grow, see them change their behavior based on what you do with them.
There is something really neat about it. I mean, again, I'm not doing anything like that. I've got a slime mold that I'm trying to culture right now, but that's I'm doing it in an egg carton, so it's not really the same. Yeah, not quite the same thing.
You could do a sourdough. That's similar.
Sourdoughs, yeah. I mean, that's. Yeah. I mean, well, now that's interesting. Talking about beers and stuff. Are you doing fermentation and stuff as well? Are you interested in all that? No. Yeah. No, that's a whole world that I feel like I would love, but I really don't want to get into because I think I'd be sticky all the time.
No, a friend of mine actually did it a couple of times in his basement.
Yeah.
And pretty good beers, I'd have to say.
Yeah. It's funny. I love the science behind it, and I love the idea of it. But it's one of those things I don't feel like I could invest the time and energy. I don't think I've got the patience for it. Oh, that's a good question. Do you have the patience? Are you obviously this is or do you find yourself like, come on, or is it?
No, I think I have the patience for it, for those types of things, because I know you need the patience and you can't rush it.
Yeah.
But in general, sometimes I don't have patience for. I'd say I don't have patience for stupidity, but who does?
Yeah. It's definitely.
Sometimes when people, especially the higher ups, like, yeah, but why can't you do this quicker? Because it says in the protocol the incubation time is 30 minutes plus 25 there. Then you'll be done within an hour.
Right.
Like, yeah, that's not how it works, because you do have all the manual labor in between to prep it for every step of the way. It just takes 2 times extra. Don't have patience for that.
Is there stuff that you can, I mean, we're talking about big data, we're talking about these tests and obviously these complex machinery. I mean, how much of this stuff is literally hands-on and how much of it is some sort of robotic system? I mean, is it literally pipettes and
Well, the stuff we do is, I'd say, 95% hands-on.
Really? Okay.
We do have some of the flow cytometers where you can just put in your sample plate, so it has 96 wells in it. It's like small reactor volumes, and then the machine goes from A1, A2, etc., all the way to the end.
Oh wow!
And you can automate that, but there are a lot of companies or research groups who do use robotics for all their pipetting, but they're more involved in mass screening, compounds,
yes,
or other things and conditions, etc.
Whereas you're focused on, a few specific.
Yeah, we're far smaller scale, or our experiments don't really lend well to automating it because it is so specific.
Right.
Yeah.
So your job is safe for the time being.
For now, yes.
What are you working on now? What's the project? What have you got? Like,
I'm doing a lot of stuff for colleagues. Couple of them are, of course, when they're on vacation, I take over their research, but we do a lot of cell culturing with the ATO and the ABO system. I'm not sure if you read the papers on it.
Like I said, I had
you skimmed them.
It was quite a skim. Quite a skim.
That's fine. Oh, we had the ATO system. It's artificial thymic organoid. So organoids are small, small cell clumps.
These are the thymus. Your thymus.
Yeah. We try to mimic the conditions that are present in the human thymus. And the thymus is one of the organs where T cells, so part of the immune system I explained earlier, develop.
Behind here.
Yeah.
Very
good.
That was.
Did Frank. You know you can?
Frank told me.
Yeah, Frank is much better at explaining the theoretical parts than I am. But
he's great. He was a great chap. But I'm enjoying this too. So yeah.
Yeah. Good. That's good to hear. No, we make those artificial thymic organoids with CD34 positive cells, and they are
wow.
CD34 is one of the markers I explained, the proteins that are on the cell surface, and cells that are you can take them from, let's say, cord blood, so from a freshborn baby, or from bone marrow. You can isolate those CD34 positive cells. You select on them. We take those, put those into culture combined with a stromal cell line. In this case, it's mouse. We haven't got a human to work yet, but we're working on that. And those mouse cells you combine them into a very small blob from 5 microliters, and you take a couple of 100,000 stromal cells. You combine them with a couple thousand of those stem cells.
It is a
recipe. And you put them on filters with some culture media beneath it, and then they start to slowly develop and grow, and those stem cells start to differentiate into the T cells or the B cells for the ABO system. And depending on what type of cells you want, you can give them chemical compounds to push them in the right direction.
So are they pluripotent, or are they, or there's 2 different terms? There's one that you can force into different things, and there's one that just is.
Yeah, you have pluripotent, you have multipotent. Oh, I always get them confused.
Yeah, me too.
But, these are a specific type, so they're not the actual stem cell that can go into everything. These are already primed to go towards the immune cells.
Okay, all right.
They're already
geared towards that.
They have gotten nudged a little bit already.
Right. And is that something that's provided for you, or do you have to create those yourself?
These stem cells we isolate from cord blood, mostly what we're working with. And we have, it's called the geboortehuis in het LUMC. I'm not sure how to translate it to English. Pretty much, it's pretty much a birth house where all the pregnant women who go into labor, go there. And then they can choose to donate the cord blood.
Right. Okay.
For science,
which of course is rich in stem cells, I'm guessing. Yeah.
Yes. Okay, and the younger you are, the more rich your blood is as well. So the older you get, the less and less you have them in your blood.
Wow.
Which they think plays one part in the aging.
Yeah. That's interesting. So, so is I'm wondering if this is the same. Is it the same in North America? Or I feel like we're shying very much away from the cord blood stuff. We seem to be trying to come up with other ways of forcing things back into pluripotency, as opposed to, because of all of the politics around it.
No, we're definitely working on that as well, and we have iPSCs, we call them, which are induced pluripotent stem cells.
Yes.
And you can make those by treating other cells, which are isolated with a specific set of compounds. I don't know. I don't do that. But we take those and we grow them out, and then we can nudge them in the proper direction to make the immune cells as well.
It's amazing. It's amazing because it's like this is real. I mean, this is science fiction to me. It's the idea that we can take these cells and turn them into, and we can decide what they're going to become. There was an amazing quote that someone said about that we are now in charge of our evolution. It used to be our environment, but now basically we are the ones who will now steer the direction.
No, we definitely are.
For good and bad.
Yeah, the things people can now survive, which we figured out we couldn't, like, say 100 years ago. It's insane.
Well, childbirth being one of them.
One of them.
I mean, that was the extraordinary fatality rate of childbirth.
And one of the reasons we have such a difficulty with giving birth is because we started to walk up straight. But we managed to figure it out to reduce the risk, and then you don't get selected for it anymore. So
interesting. So we were crawling around on our hands and feet. Then it was an easier process. We're designed for that. Wow.
It's also one of the reasons why babies are born so, like, not complete. I was gonna say, how do you call that?
I don't know. What do you mean?
If you compare it to, like, a giraffe baby, once a giraffe baby is born, it can instantly walk. For us, it takes at least a year. We're underdeveloped. That's the word I'm looking for.
Interesting.
We get born reasonably underdeveloped.
It seems like a really bad evolutionary choice, because I feel like you're just a target.
True, but we settled down already, and we had the baby survived, so that trait never got evolved out of it. It stuck around.
Interesting. Well, there you go. So maybe who knows? Like, I mean, they design easy care sheep that have heads that are easier to birth. Like they literally. That's like there's
Don't give them ideas, though.
I know. The next thing you know, we're all going to be cone heads, and
that's an old school movie. I haven't heard in years.
Yeah, it's a reference. I don't think I've actually ever seen it.
It is funny, but definitely a product of its time.
Yes, I'm sure. Yeah. Not a lot of these things age very well. Okay, so I've taken us completely off track now. But the stem cells that you're working with, you are using them to create these basically organs on a chip, organs in a plate type thing. Organelle. I guess.
It's pretty much on a petri dish, I would say, and that petri dish has an insert with a filter on top. So
what does the filter do?
It prevents the organoid from drowning in the media, the growth media below it.
Oh, so it's like a plant. You're like hanging it over the water.
It's like a sort of air liquid interface where you have the organoid on the filter, and then below you have a bed of growth media. So it gets its nutrients from the bottom, but from the top it remains relatively dry, I would say.
Interesting. Well, and that's just something trial and error. You figured out that's the way it works.
Yeah, well, we didn't develop it ourselves, but that's how they figured it out. That's the best way to grow them so far.
That's got to feel pretty good. Like you got to feel pretty good coming in the office and going like, oh, look, it's a little thymus. Like, or could you do this with other? I mean, I know they've done brains on chips and things, or not brains on chips, but they've done certain neurons and stuff. Is there?
Yeah, they're doing it with neurons, for sure. Groups in our department are working, I think, on organoids for
like heart
for your gut endothelium.
Oh,
they study Crohn's disease and stuff like that.
Oh. And the advantage, I guess, is that you're experimenting on humans without experimenting on humans, and also
it's
not mice.
Yeah, you're kind of bridging the gap between fully mouse model up to fully humanoid.
But how how good is that? Like, I feel like, isn't there generally an element of like, it's when everything gets together in that squishy, when it all squishes together, that's when a lot of things can change. Are you getting, what do you get out of these thymuses? What are you getting?
It gives, of course, it's a model, so it's never 100% representative of what happens in the human body, but it does give us a good indication of how T cell development works, and then we can apply that knowledge to further studies or see how we can nudge T cell development in a different way, or where does it stop? We have some essays running where you modify.
What's an essay again? When you say essay, what is an
essay? Is just an experiment. I would call it. Yeah.
All right.
Not
I like to know
essay.
the lingo. I want to get the lingo.
Yeah. Yeah. It's not like an essay you write in a lab.
Yeah. I was like, yeah, that sounds awful. Yeah.
But yeah, we run those, and we can of course modify the T cells in our lab with viruses or other means, and then we can prevent them or give them a defect and see how does this affect development. So let's say you knock out a gene so it doesn't function anymore. How does that have an effect?
What's the knock-on effect of that?
Yeah, knock on or knock out, stuff like that.
I guess that's true.
So with the RAC1 trial, like that's the disease we're working on, the gene therapy.
Yeah.
It has a mutation in their DNA where the gene doesn't function, or the protein that comes from the gene doesn't function properly anymore. So you have
the bubble boy.
Yeah, exactly.
Yeah.
So you have the aberrant T cell development, and your immune system doesn't work. We can induce similar mutations and then see how that affects the development of your T cell, and then once you do that, you of course gain more knowledge on how does development actually work. Yeah. Can we fix it?
You break it so you can fix it.
Sort of. It's like breaking things to see how it works. Sort of like
reverse. I'm very good at breaking things.
Reverse engineering your immune system, I would call it. Yeah.
Well, that's
a good way to describe it.
I think it was Feynman who said that you don't understand something until you can build it, which I thought was kind of a neat way.
That's a very, very good way of putting it, I'd say.
He was pretty smart. Yeah, I got the sense.
He's a smart one.
Yeah. But you've got there is a gene therapy that basically fixes this in kids at this point.
Yeah, that's what we're working on in the lab, and we have a clinical trial running where we have some kids treated with the product.
Do you ever meet the kids?
No, I don't. No. We do get a lot of samples, and I think, of course, my boss and the doctors involved meet them.
I'm not sure I'd want to. Like, I feel like like, oh God.
I'd probably get too
kids.
Attached.
Yeah.
We sometimes have to go to the pediatrics department to pick up some material, and then you run into all those parents with their sick kids, and it's heartbreaking.
Yeah.
It's not nice.
It's just brutal. Yeah. But what you're doing, of course, could completely change. I mean, there is a chance that what you're doing could lead to some kid coming into your lab as a student who's been
potentially
saved by this.
Yeah. It's going to be a couple of years, but I think the oldest one in the trial is about 7-ish.
7,
I'd say.
Wow. Yeah. You'd be amazed how fast it goes, though. I had a kid. Well, Jane had a kid. I watched, and all of a sudden they're like 19. It's bizarre. It just happens.
Yeah.
You got kids?
No. Oh, I have a dog.
Hold off as long as you can. That's what I generally.
We're not starting, if we even want to, until my wife finishes her PhD. So
smart. And the other thing is, until you've seen every movie you ever want to see. Because once you have the little kid, forget it. You're never seeing a movie that doesn't involve like an orange, purple dinosaur in it or whatever. Yeah, it's terrible.
One of our good friends has 3 kids, and he's always tired. So
3 I don't understand at all. 1 is so perfect. The thing that got us was everyone says like, oh, the first kid is one thing, and then the next kid is completely different. We're like, you know what? We like the one we've got. We'll stick with that.
Fair enough. Fair enough. Yeah.
What's your wife studying? What's the?
She's in the dermatology department.
Oh wow!
And she is investigating cutaneous T cell lymphomas.
Okay.
Which is a very difficult string of words. I get that.
But that's the T cells are back in there.
Yeah.
Did you meet over a?
Yeah, we met at our first job.
Did
in
you?
the same lab. Yeah, we did.
Good. You did.
Yeah.
You met over a Petri dish of T cells.
Yeah. But no, she moved over there to do her PhD, and it involves a lymphoma, which is a type of cancer, and develops, I think, if I say it properly, it develops in the blood and then migrates to the skin.
Oh, weird.
And causes severe reactions.
God.
So it's not pretty, but it is luckily very, very rare, and mostly occurs in older people.
Oh, okay. All right. Well, they say you want to know how to get cancer, just get old enough.
Basically. Yeah. Eventually, if you don't die of any other cause, you'll eventually die of cancer.
It's like a default. Yeah. Okay. And so where are you at with your thymus? You've got clinical trials happening, which are, by the sound of them, doing very well.
Yes. For this one, so far, all the inclusions are alive and well. So.
And you're just continually trying to get better at it, or why continue with it? If you think you've got it solved, what's to change?
Yeah, I think you need to gather a lot of data and trials to actually see if it is safe. And I think this is the first step towards commercializing it. But I'm not going to say.
It is so frustrating that money is involved. It drives me absolutely insane.
Yeah, it is expensive, and I get why it's needed, but it is frustrating. I can't say too much about it because then I'll probably get stuff wrong, and I don't want to do that.
Right.
But it is very.
They'll take your thymus away, and then that's not.
It might help applying the knowledge we get from this, so the techniques, etc., everything involved, to also use it for different diseases that have a similar genetic defect.
Right.
So if that works, if it works here, then we can apply it to other types of defects.
What else? What other ones have you tried? Any others specifically, or?
I think I'm not sure if I'm allowed to say that.
Oh, then don't.
No. I don't want to ruin everything or give other people
I'd rather
ideas,
you
right?
still had a job at the end of this interview. That'd be nice.
Nah, it'll probably be fine, but there are similar defects we're working on.
Right. Okay.
Related to different types of immune cells.
Right. That has got to feel pretty freaking great. I mean, how do you keep up on this stuff? Are you reading papers all the time and stuff as well, or
not as much as I should? But I focus more on the lab.
Everyone says the same thing. Every single person I've asked says the same thing. Yeah.
I'm a very slow reader, so
so am I. I know it drives me nuts.
Yeah.
Do you use AI?
No, I refuse to. I'm not a fan of it. Are
you an organic intelligence?
No, I am. Instead of artificial intelligence, I always say I have genuine stupidity. So
that's a better T-shirt for me,
right?
I should probably get that. I'm curious because I absolutely love it because again, what I'm dealing with is I'm not dealing in absolutes. If I get stuff wrong, it's like someone goes like, "No, David, you got that wrong on a podcast." Yeah. So I understand that it's not. But from an educational perspective, for me, I'm learning stuff and have time to understand stuff that I would just never be able to do. I don't think I'd be able to do this show without it.
No, I'm not as opposed to it as I just made it out to be, but
some people are like it's very.
I think the general AI we're working with, let's say ChatGPT and stuff like that. I think it's not useful for the investment that it needs. I am a fan of AI, where what they used to call, I think, machine learning or models, where you train an AI on like a massive amount of input for, let's say, MRI scans, like the data that comes out of a flow cytometer. You can there's a lot of diagnostic
pattern recognition.
Yeah, exactly. For a lot of software for that, and it's very useful for that. It can detect very minor defects or changes in the standard, and then the energy, etc., it takes to use it for those applications. So very focused and specific applications. I'd say it's amazing for that. And I'm happy. I'm really excited for the developments in that field. But I just think the general AI as in how it is now is, first of all, making people stupider. I'm absolutely convinced of it. Seeing as how many people use it. Not everyone, and a lot of people use it very properly. Don't get me wrong on that. But yeah, I think so far it is not in a state that it is very useful currently.
It worries me. and it also and inspires me, because I feel like if you are going to school to get a mark and a diploma and a job, and you're using AI for that, I worry about that, because I feel like then it's truly just doing the thinking for you. But if you are someone who is curious and passionate about what they do, it is a wonderful tool, because it complements all of your curiosity.
No, you can definitely use it as a tool to get more suggestions on what you should read next or where you should focus on next. Definitely, because it knows a lot. But it is scary as well, because it is very well known currently to have a lot of errors in it.
Yeah,
And
absolutely.
in a detail based field, it is very tricky and very dangerous.
That's where you and I. That's why we're in a different world. That's why it works so well for me and probably not for you. Is that for me, the specifics doesn't matter to me. I just want a general understanding of stuff. I will still go. I must say. I mean, like I say, I still go to YouTube and watch people explaining. God, what's it called? Flow
cytometry.
Flow cytometry. For some reason, that is a killer for me. We should get to some questions too, because I realize we've got questions, and we've just been talking for how long? We've been talking for over an hour.
Yeah. I told you I started rambling when.
No, it's not
when
you.
It's going.
Like this is just this is I don't know how you're gonna get any work done if I ever come and visit. You're saying I can actually try. I could maybe build a thymus or something while I'm there.
Oh, that's going to be tricky. We have some projects working on it, like growing a thymus from stem cell up. We're not there yet. I'll tell you that much.
Oh, okay. All right.
I can definitely let you make some ATOs or ABOs and do some pipetting.
Okay, great. Perfect.
Show you some stuff under the microscope.
Oh, see, I love the microscope. The microscope is just like, whenever we go up to a cottage every summer with some friends of ours who are entomologists. So it's wonderful. Like you go and there's, they've got binoculars that look at insects, so they could stand, a foot away and zoom in on an insect. It's just
still amazing.
Oh, it's extraordinary.
It's not my field, but I'm happy that people are doing it.
Oh, it's wonderful. And also just the latest thing she was doing was they were doing she was a science consultant on a show that's dealing with bites and stings, and how we're using those venoms and and salivas and things, obviously made famous by the Gila monster with Ozempic and stuff. But the idea of using these toxins and poisons and things in nature to cure things and to do everything from erectile dysfunction from spider bite. Like literally, they're using spiders to cure that. And then
it makes sense, because a lot of those compounds are used to, let's say, dilute your blood so it keeps flowing even though you have a wound, so they can feast on it. You can isolate the compounds responsible for that, and then
clot. Or they'll clot really, so they've got
clotting the other way.
They've got a material they pulled that allows a wound can clot like that.
A funny, let's say, fact about that. About anticoagulants that we use in tubes to collect blood. Have you ever heard of sodium heparin?
No.
That's one of the agents we use, and heparin was actually first
that keeps
isolated.
It from clotting while it's
so anti-clotting. It's the first. I think it was isolated from a snake bite and the venom from that. And that's how you get that's how you get from nature to applying it.
Nature's done it all. We're just trying to figure it out. We just got to, like you say, we got to figure out. We got to understand it so we can build it ourselves. That's all.
Exactly. Build it potentially better.
Is there anything when you're not working? What are you doing? Do you have your own little science experiments of your own, or you just avoid it entirely? No. What's your? I try
to think as little as possible about work when I'm
really
home.
Yeah. So what do you do? What's the? How do you relax?
I still do. I play video games, read a lot of science fiction and fantasy books.
See, you said all the right things. Okay, what games?
I do some Gunpla building games.
You do some what?
Gunpla. So have you ever heard of Gundam, the animation series?
Yeah.
There's a lot of models you can build, like plastic model kits.
You're looking around. They're all around you, aren't they?
Yeah, I'm trying, but we recently had to clean our house because we were selling it. So I stored them all away.
I would say death, divorce, and moving. 3 most stressful things you could possibly do. Yeah.
Yeah. Well, I'm not planning to do the death and the divorce. So
yeah. It's amazing how often it leads to that, the moving part, though, because moving is just so stressful. I hate it.
Yeah. I get that, but
where are you going? Where are you moving to?
Buitenkaag.
Where's that?
From Oegstgeest to Buitenkaag.
Wow.
Yeah, difficult.
Neither of them are easy to pronounce. Yeah,
they're more difficult
near Leiden?
than my last name.
Still near Leiden?
Yeah, Oegstgeest is. I am at a 5-minute bike ride from Leiden currently.
Oh, nice. Okay.
And Buitenkaag will be about 30 to 40 minutes ish.
Oh, really? And the advantage being you are closer to or further from.
No, we got a house there. It's a bit of a difficult housing situation in the Netherlands.
Oh, is it? Is it expensive or is it just not available?
Yeah, both.
Okay, right.
But we had the opportunity to join on a project that was newly being built. So we got one of the houses there.
Oh, nice.
And it's still relatively close by. So.
And now you're on the property ladder.
Yeah. Well, we already owned this apartment.
Oh, okay.
So we were luckily already on it. We got very lucky.
I mean, you kind of, unless you're on it, I don't know how people. My son is.
Me neither.
He's just going to have to knock
the old house I was living in before this is, I think it's gotten twice as expensive in the last 6 years or something.
Yeah, it's about the same here. It's about doubled since we moved here. It's extraordinary. It's great if you own a house, but then it's not so great if you sell it and get another one because you still got to pay twice as much for a house as So that's my plan. My plan is sell here, go back to England where my wife has her dad's place.
Okay.
I get a pig and a tractor, and I just come and visit you in the lab all the time.
I was going to say you were closer by then as well.
That's the advantage. It's a better jumping off point. So we'll see. But I should go to some questions because I feel like I've completely monopolized you. Is there anything? Well, no. We started this, and then we forgot. Okay, so you're building these models. What do they call it?
Yeah, it's more like assembling. It's not actually building it. You get them in large plastic trays, you call them, and you cut off the pieces. And
I love it. You know what?
Yeah.
I just 3D printed some. Hang on a second.
Really?
My son is working at a camp, and he's got a lot of kids who are sort of on the spectrum, very, it's kind of somehow difficult to manage and stuff. So we started 3D printing these little.
Oh yeah, they're exactly like those trays we get, and then I cut the parts loose and I assemble them. You
got a 3D printer?
Nope.
Oh, dude,
I don't have the space for it yet. I'll get it eventually.
Yeah, I'm telling you, if you like that kind of stuff, it's like a dream factory. I'm 3D printing glasses. I like these. No, not these. Not the other glasses. The other glasses are 3D printed. I love. It's amazing. All right. Okay. So, I never had the patience for that stuff, for building the models that were.
It is very relaxing.
Is it?
Yeah, it is.
Okay, all right, I'll give it a go.
But
I guess if you can just focus on it.
Maybe not to you, but yeah.
Like I say, I'm also better at breaking things than I am at actually building them. Okay, so the model building stuff. You've got what's current any sci-fi stuff right now? What are you watching right now? What's the go-to?
For TV shows, I picked up For All Mankind. I started that. I'm not very far in, but
getting recommended to me.
Yeah, I heard it's so far it's very good. It's very slow.
Yeah,
in a good way. In a good way. Yeah.
Believe me, I just watched The Odyssey. You don't. It's that's slow.
Yeah, fair enough.
Okay, so For All Mankind, did you get into like Foundation or Silo or any other ones?
No, Silo's on my list. Foundation, I quit halfway through because I read all the books and it deviated too much from my liking.
I didn't read all the books. I only read one, but I kind of want to go back and read them now because I just love that concept of this sort of mathematical model. It's just wonderful.
They're pretty good. Yeah.
Yeah. I vaguely remember, like I read Foundation long before I could understand it. I think it was more, I'm always reminded of that line from from Fish Called Wanda, where it's like, monkeys can read it, they just don't understand it. That's kind of where I was when I read. So I feel like I should go back and revisit that.
I have that with a lot of stuff still.
Yeah. So, okay. So anything else? Are you a Marvel fan? Do you like all that Hollywood stuff?
Not the newest stuff.
Yeah, I mean,
and I know that's gonna seem very how do you call that? Like, everyone says the newer stuff is shit, but
I'm with it. I've kind of tuned out Marvel now. I don't even bother. I'm looking for. I'm actually watching more horror now because I find it more exciting to me. Like there's more sort of like, oh, is that Whereas I find a lot of the science fiction, because it's expensive, it has to sort of appeal to everyone, so it appeals to nobody. Yeah, people do say the new Spider-Man's good, but I don't count that as science fiction. I don't like, that's not really sci-fi to me.
No, that's fair. No,
sorry, that was a chat. That was a chat thing that popped up. So.
Oh, okay.
You don't have to read the chat. It's fine. We're gonna do questions.
I have
now.
the YouTube chat next to me here. So.
Oh, do you? Oh, that's oh my. Well, then you're already better than I am, because I can't. I get distracted by that in a second. I'm gonna go. So what we've done is my fabulous friend Susan is here and taking questions. But before we get to the questions, I have to say a huge thank you to the Patreons, and specifically to our 5 co-geniuses who have actually specifically become patrons for this show, to make this show happen. So, and I love their names too. There's Psycho Liz, always a good sign. Thank you, Psycho Liz. Thank you, Samantha V, and Minirockhopper, which for some reason always makes me think of Hail Mary, Project Hail Mary. But Minirockhopper, Ryan, and Jenn, thank you so much for being co-geniuses. I can't tell you how much I appreciate it. It's fantastic. And of course, a huge thank you to Susan for sorting all these questions out, and just being so wonderful in general, and Agne for making it happen in the first place. So, and I saw Cal and Jack in the chat. So welcome. Thank you again, Cal, for probably controlling for being the hammer in our chat, making sure things stay on track. All right, questions. Oh, look at that. Jen the Nerd says, silly question. Worst and best Star Trek Voyager characters to be stuck on an island with?
Oh God, I don't think I even remember all the characters, but difficult.
I can't believe you got a Voyager question as the first one. I've never seen it.
I don't think I want to be stuck on an island with Neelix. Then again, you'd have a free food supply because he'll figure something out. Or, oh God, what are all the characters called?
Yes, you get no help from me because I haven't seen it.
No, it's been so long ago that I watched Voyager.
Right.
Then again, I probably would be happy to be stuck on an island with Janeway itself, because she'll figure something out to get us off of it.
Yeah, you kind of want someone smart who can figure out how to get you out of it. I'm sort of with that. Yeah. Why? But I like though, the idea is not to enjoy your time on the island. It's just to get back off it again.
Yeah, just get me off of this.
That's your approach. I don't want to be on an island. I like being in the lab. Yeah. Okay, Wholesome Vampire says, Wholesome Vampire 24/7 says, what was the most interesting project you remember working on?
Well, the gene therapy project absolutely still is interesting. Yeah. And maybe my very first project I worked on during my bachelor internship, where we worked with immune cells again, but in mice, and we had cell-specific knockouts. So we modified some cells in a different way that the gene was only knocked out in specific types of cells. So the T cells would have it active, but in the B cells it didn't work.
Huh. And what were you trying to figure out there, or what did you figure out there?
How it affected the development of atherosclerosis, so the plaque formation in your blood vessels.
Oh wow!
If it alleviated it or made it worse, depending on the diet.
Wow.
Oh.
And what did you figure anything out of there? What did you get?
We did have some very interesting and promising data, but in hindsight, it appeared that the knockout wasn't very successful, so we could throw everything in the trash.
Would that be counted as the the negative information? What would you call it?
Yeah, in a way, you could still. Yeah, I think you could still use the results as a follow-up for a study that was properly designed and prepared. So you do have some ideas on how to work and continue your studies, but the data itself was useless in the sense that it didn't tell you everything, because the basis of the material you work with wasn't up to par.
Was that just a matter of just you guys not knowing enough about it, or is it just the state
we got the knockouts from somewhere else, and I don't think they did their controls properly. Check their work properly, so that was unfortunate.
Check your works, folks.
Yeah.
Is there anything, like, if you had your druthers, if you were an unlimited funding and stuff, what would you be doing now? Would you have something specific you'd be focused on, or this is it?
I don't think so. That's also why I didn't do a PhD or postdoc. I'm not creative enough to think of my own research. I'll admit to that.
I think it's a different type of creativity. I think that's the key. It's a different, I think it's you don't get a lot made without the technicians, and you don't get a lot made without the people coming up with their crazy ideas, right?
No, definitely, you need the combination of everything.
Yeah. Okay. Next question is that's so it's basically the question I just asked. Was actually, I think I've just beat Susan to her question. But if money and politics weren't factors, what would you love to see happen in your field? Sort of along the same line, but yeah.
I would say research would focus more on rare diseases, because now everything is cancer. Cancer is the hype.
Yeah.
Everything goes towards but what type?
Because there's money in cancer.
Same with when COVID happened, which was of course horrible, and it was good that it happened, but suddenly everything went into overdrive and all the money went to that.
Yeah.
There should just be more money in science in general.
It's funny. The thing that the weird sort of epiphany that I came to while talking with Frank when we did our Co-Geniuses with him was I don't we made up money. Like, we made this up. This is a concept that we have come up with ourselves. Yeah. It's imaginary. Life and death isn't. But for some reason, we are now putting the two together and going like, okay, is it worth the money? It's like, of course it's worth the money. It's not. Yeah. This money's,
How can you put money to worth monetary value to a life?
It doesn't make any sense. I mean, forget even from just like an emotional level. Like, I mean, just purely from as you say, from a purely sort of like logical or philosophical level, it does seem like we're selling ourselves short by focusing on that, right? There's a wonderful quote at the beginning of Hitchhiker's Guide to the Galaxy, which, if you haven't read, have you read it? Love it. Where he talks about it's a planet where people are obsessed with passing little pieces of green paper back and forth. And it was, of course, that was before we went digital. It was money. It was money, and it was just that wonderful way that he reduced our obsession with this to its stupid, absolute stupidity level of what it actually was. We were literally passing paper around.
It is kind of,
yeah. Anyway, that's not to steal your question.
No, don't worry.
So instead of reacting in a political way or in a sort of, oh my God, it's over there, why don't we just constantly spend this money and throw it into the science research stuff?
Yeah, I'd say we need more money in science in general. And then if someone comes up, let's say you have I want to research this field. Okay, here's $100,000 or something to get you started. And if it's
so, who does that? Is that a government thing? Is that a public thing? A private thing? What do you think? Where's
both? There's both. There's like the charities, and there's government funds. And you can write to them with your proposal. Let's say, I have an idea to investigate male pattern baldness. Which,
I don't know why you would say that. What are you talking about? I've got headphones.
I see myself, so that's why. You're fine. You're fine.
Yeah. I don't know. I feel like it just goes further and further back.
But it is a very easy thing to say. And then someone goes, I have an idea for that. And you have to write your proposal. I'm going to do this research, include this, and that, and try it and figure it out. And then a committee judges your proposal, and it says, okay, here's the grant, and how much money do you need? And then you get a part of it or all of it. And then there's charities that are specifically focused on cancer or on like your gut, your bowel system.
Yeah.
And then you can write to those specific ones and then get money from that.
The weird thing I'm seeing now, though, is that people seem to be so distrustful of this stuff, and yet at the same time they're like, so I'm going to make my own Ozempic, or I'm going to get my own peptides or whatever it is that I'm going to be using, or my own supplements and stuff. I don't trust the man. I'm going with the dodgy stuff. It's like, do you think is that just a lack of education? Is that
yes.
Okay. All right.
Yeah.
It really is.
Lack of education is a serious problem, and it's getting worse. Combined with
Are you seeing a difference I mean, not to put any of your students on the spot, but are you seeing a difference in people's approach as they
not the students we get are all higher education.
Right. They've been picked out.
And that'll stay the same. But the lack of education, I'm more implying that like all the budget cuts, etc., are giving people less opportunity to get an education. So you'll never see those people, and they don't get you can see it happening everywhere, and it's the easiest way to get budget cuts for the government as well. We'll just cut education, or we'll just cut healthcare.
I never understood why those were the ones that went. I was like, what about,
No.
I don't know, political salaries?
and not even that, because you want your politicians to be compensated, otherwise they won't do the job. But it's so much easier to just tax the companies, like the big ass companies who don't pay anything.
Yeah,
but hey,
I mean, it does. Yeah,
I have opinions on that. I don't think this is the place to share that.
Yeah, probably. Yeah, it's not. We generally like. I mean, Agne's always very big on avoiding politics, and I agree with her. I think there's a this is also, I think this is about celebrating. This is about celebrating science and technology and the people who do it, and also introducing people to stuff that and to areas of science or even jobs that they probably didn't know existed. Like, I mean, I never would have. I mean, it's not that I wouldn't have thought of the technician side of this part, but I don't think I would have understood exactly what it was or what it entailed. And, it's.
I think a lot of, you can compare it to the, like the CSI techs.
Right. Yeah.
They do that's a good comparison. I don't know if you ever watched the Flash.
Oh yeah. I watched the first
season. Out as a lab technician, but for forensics, but we do it in like medical science. Right. That's a little bit of a comparison you can run.
I like that. I mean, that makes sense though, because you're doing the same. You're looking I mean, you're basically looking for what cause of death and any trace that. I mean, that's interesting.
In a way,
it's. In a way, it's the marketing of education, I think, that's tough as well.
Education is not fancy or glorious or and that is
the problem with learning is it's hard and it's work, and that's the very nature of it. I think the fact that you overcome an obstacle of understanding, so to speak, that overcoming, you're an idiot, and then you're I'm a genius. Like it's that moment. That's the learning part. And the problem is we've spent so much time trying to take that away, and I think AI could potentially take that away from people as well. Where then you don't learn, you're just looking it up, and then you lose the sort of where's the intuition, where's the the human part. All right, wait a sec. So another one from Susan. Okay, what helps keep T cells you're working with stay on target for specific cancer cells versus traveling to other parts of the body? What keeps on target? Oh, right. This is the thing we haven't really talked about, but you're also one of the other things you've been doing is looking at a blood test that has a fingerprint of cancer on it, basically, that you'd be able to detect cancer from a blood test. Is that?
Yeah, that's the idea, because there aren't very many tests that can identify cancer in the very, very, very early stages.
Right.
But your immune system is already working to process those tumors or cancer cells.
Right.
And what they wanted to do there is develop a blood test, so those cells go there, they eat a part of the cancer, and they process it internally. Like chop it up pretty much to small parts. And what we wanted to do is see if we can draw some blood, isolate those cells, and then isolate or sequence the smaller parts of the tumor in there.
So you could identify that there was a tumor before you'd know there was a tumor.
Yeah, either identify it, so for early detection, and even further, which is very much further, you can use those parts that they chopped up and then specifically target those parts of that tumor cell.
Oh, nice. So it becomes almost like a marker for the tumor, and then you sort of stick the immune system on it yourself.
Yeah. That is one. And of course,
how far along are we with that?
Not very. It's very difficult. Yeah.
But it's
sad
possible
to say, but it is potentially, yes, especially with how the techniques are developed. Sensitivity increases. You can get there. You can eventually get there.
And hopefully. If it's like my lack of knowledge of this stuff is obviously going to show here, but if it's showing up in your bloodstream, does that not mean it's already quite pronounced, or you're saying it could be?
No. It could still be like very localized, but the immune system has already detected it and then destroys or eats some cancer cells. It doesn't mean it has already metastasized.
Oh, okay. All right. So this could literally be like better.
Yeah, it could just be like how cancer starts. It's one cell that gets a defect either by UV radiation or some other compound that happens, and it changes that your immune system detects it, eats it, and then you already gets rid of it. But sometimes it escapes it.
So
and even though it escapes it, your immune system still will eat parts of it, and then you can detect it.
Right. But then you'd be detecting, but then the next problem would be, how did you detect the ones that are a problem rather than the ones that your system has already dealt with?
That's a good question. And usually the differences are very, very, very minor.
Yeah. That's the problem with cancer. I feel like cancer is the same problem as AI, in that AI is being used to describe such a huge, massive, different number of different things. Cancer is kind of the same thing, isn't it?
Yeah. Cancer is like a blanket term for
I feel like
so many.
Yeah.
You already let's take breast cancer. There's already, I think there's 10 different versions of it. Lung cancer, blood cancer, bone cancer, brain cancer. There's very, very many different types of it.
What were you looking for? What were the markers? Were there a specific cancer you were looking for the markers?
I don't remember specifically, but it had.
How long ago was this you were doing?
A couple of years.
Yeah, okay.
I think 5 years ago or something. But it had to do with, I think, intestinal cancer.
Oh yeah, right. God, yeah. That stuff just terrifies me. That whole region of going wrong is just not something I want to know about. It's bad enough, with a dog picking up after that. I don't want to.
Yeah, I get that.
Don't get a dog. It's my other advice.
I have one. So
do you? What do you have?
Yeah, I have a doodle.
A doodle.
Yeah,
I have.
She's very cute.
Yeah, they're they're lovely and cute until it's 6 o'clock in the morning. They want to go out. Yes, I love my dog. I just Huzzah drives me nuts. That's all. That's why I called her Huzzah. Okay. Are you okay for time? Let me know. Are you?
Yeah, I'm fine. I have all the time in the world.
Okay. All right. So, I went off topic for that because the question was, what helps keep T cells you're working on stay on target for specific cancer cells versus traveling to other parts of the body? So once you've identified these things, that marker is what would draw them to the right part of the right cancer?
No, not specifically, but I'll have to admit I'm not as well versed as I should have been in this, and I don't know most of the theoretical parts. But in general, what you have these things called signal molecules, cytokines, etc., and they are excreted by cells, and then different types of immune cells have different receptors for it, and then they detect the gradients, and then they home slowly towards those.
So those are the ones that would latch onto something and say, "Hey, over here," type thing.
Yeah, you have some of them are excreted, and then they draw other cells towards them.
Right.
Usually, they then interface with each other, interact with the surface proteins I mentioned, and then they attach to each other like receptors and. And that's how they interact together, and then decide what are we going to do with it.
Okay, so
it's very difficult to explain.
I can imagine, because the other problem, of course, is that, as you say, it depends on the cancer. It depends on and even the specific type of the specific cancer has different. Can be. I mean, the whole problem is
and sometimes even in between in the same cancer, you can have different signals.
Right. There's a brilliant video that James Gurney, our friend James, worked on for Kurzgesagt, this wonderful.
I know of them.
They have some beautiful, beautiful videos on cancer that really help.
They're very good at simplifying.
Yeah, it's fantastic. I mean, I use them a lot. They're kind of like my go-to for biology for dummies and stuff. That's where I'll go to just get a basic understanding from it. But they have a wonderful one on cancer that talks about how even a cancer is not a cancer. It can have multiple different types of cancer in it, and that our system comes and attacks it, and then it leaves a little behind, and that's even stronger now. So
I think that has to do with the thing they sometimes called cancer stem cells. So let's say you have a tumor, and then the outer layers are the cells that get sent to metastasize or are the more vulnerable ones, and then in the center it's theorized that there's a sort of like a batch of cancer stem cells that keep proliferating and then expand.
Wow.
And that's if you then you move like most of the tumor, and a little bit of that gets left behind, and that can of course grow out again.
Right. It's almost like a rapid evolution process of survival of the fittest, where we knock stuff
Yeah,
out.
in a way, yes.
And you're just basically just pulling out just the most virulent
you can compare it to if you try to grow bacteria on a plate, and then you put some antibiotics next to it, but you let it just proliferate, and at some point they'll get resistant to it.
Ooh, yeah, which is another whole issue unto itself.
Yeah, that's a whole can of worms.
Are you dealing with that at all? Is there any?
I one of my internships, I did a little study on that.
Yeah, because that's really worrying as well. Like we basically, we're running out of that's now. Again, back to James Gurney, he works on phages, which is and his idea is that they're trying to use them to attack some of these bacteria because they're we're running it because they're becoming resistant. They're resistant.
Yeah, they're like viruses that attack bacteria, the phages.
Yeah, they're amazing. Yeah. I get them confused with the macrophages, which are, again, they just eat the cells. They eat the bad cells, basically.
Yeah, I think phage is just like Latin for eating or something.
Eating, big eater or something. Yeah. Latin. Never understood it. Okay. All right. I think so we've sort of touched on that for Susan. Sorry, we couldn't be more specific. Jenn the nerd says, can you show us something you have on your desk? Ooh, there's a challenge. Show us something on your desk. This should be good.
I have my Switch here.
Ooh, nice. Which one? One or two?
There are two.
And what are you playing on that?
Currently Slay the Spire.
Oh, okay. All right. All right.
I'm not very good at it. I also have this.
Oh,
like a Lego Game Boy set. I still have.
Oh my God, that's brilliant! Hey, look, I've got Starry Night, which I still haven't built.
Oh, we have a replica of that in our living room.
Oh, do you? Van Gogh is like, I am a huge fan.
I would show you, but I can't take the webcam to the living room.
Yeah, that's right. I think we've imposed on your wife enough already with dinner.
Oh, that's fine. Yeah. Besides that, I don't.
No, that's some pretty good geek credit right there. You got a Lego Game Boy. Did you have a Game Boy? You're too young for a Game Boy.
Oh yeah, I got my first Game Boy when I was 4. Well, my parents got it and I confiscated it. So,
God, that thing was that I lusted after that thing. My friends all had. We didn't have the money for it. And unfortunately my parents weren't nerds, so I didn't. And that said, the first I bought this thing called the Merlin, which was the very early video game. It wasn't a video game; it was an electronic game. Basically, it was all done with LEDs. I had that thing apart in 2 hours. I bought it. They saved up. Nice. I bought this expensive thing, and then my parents were aghast that I pulled it apart within 2 hours. Did you manage
to get it back together again?
No. But I made little keypads to make the lights go on outside of it, because I thought that for some reason that was much more interesting to me than seeing it in the box. So yeah, like I said, very good at breaking things. Okay. I love Jenn the nerd. I think that should become a standard thing. Show us something on your desk. I think that's just a brilliant question. And I think has beautifully summed you up.
Yeah, a little bit. Well, yes.
It's beautifully summed up your downtime, if that makes sense.
Oh yeah. I have some gaming memorabilia as well, but.
Oh yeah. Now, you a collector? Like, do you collect stuff?
Only if I see something that I like.
That's my problem. That's the worst way to collect. That's the worst way to collect, because it's expensive, because you just end up buying everything as opposed to.
Yeah. I'm looking for two things I want in my new office in the new house, but I haven't managed to find them yet.
Well, what do you want? You're talking to the people. These are the people who might have them.
I want a replica of the Master Sword and Hylian Shield from The Legend of Zelda.
Oh wow! Okay,
which is my all-time favorite video game series from when I was young. Still,
you gotta start 3D printing. You can 3D print these things. You 3D print them in chunks, glue them together, nice spray paint on them, and you'd have that in no time. Okay, what else?
What I want, or
for
new office. I think that's it so far. Maybe a nice painting of something, but I haven't figured out what yet.
Yeah, painting wise, I haven't really done paintings. It's stuff. I'm terrible about stuff. I've also made the terrible mistake of getting into trading cards with no goals.
Which ones, like Magic or
Magic.
Pokemon?
I love Magic. I just love Magic. Not all Magic. I like the art of Magic. I don't like all these tie-ins. I'm not a big fan of those. But I like the game because it's incredibly complicated, and I don't understand it. My son thrashes me, but we have fun playing it. Pokemon I like just because again, like the shiny colors and things. But I generally prefer them as a game. But I can't. Once you start, I've got boxes and boxes and boxes. It's terrible.
I can't wrap my head around them. I am very bad at deck builders.
So. Yeah, I'm not a deck builder. I like playing the game. I like understanding some of the various different. Oh, hey, you back?
Heading out.
Oh, heading out. Oh, okay. All right.
Love you.
Love you. Say hi to Kana. It's my wife going out to be social. Apparently, there's a world out there, boss.
Yeah, I'd rather not go there either.
But, yeah, me neither. It's like dragging me. Kicking and screaming unless I have to. Okay. Again, Jen, thank you. Amazing question. Okay. Might it be possible for the work you're doing to be applied in additional ways to reduce excess T cells in people with autoimmune diseases? So is there a way to... So if you have too many T cells, there's obviously ramifications to that as well.
Yeah. It is tricky, but yeah, I would say there is an opportunity to do that, because once you know how to properly get the system running, you know which pathways or which compounds make the T cells or let.
And is that the thymus? That's coming from the thymus? Mostly,
yes.
And the bone marrow.
That's why they're called T cells. That's because they develop in the thymus.
No.
Yeah. And they're called B cells because they develop in the bone marrow.
No.
Yeah.
Oh my God! Why did
it's so easy?
But oh my!
I didn't mention that. Sorry.
No, it's not your fault. Yeah. I've been reading about this for years. No one's ever told me that.
I think that's the reason. At least.
Well, even if it's not, it doesn't matter. That's how
it works, right?
And what's the difference between the B cells and the T cells then? So are the ones that come from the bone marrow different than the ones that come from the thymus?
Yeah, T and B cells are very different, but then again, also quite similar. But they perform different.
That's a scientist answer right there.
They just perform different functions of the immune system.
Okay, all right.
B cells are more like T cells. Recognize them a bit quicker, and then you have T cells, different kinds of, and usually they recruit the B cells to help them exacerbate the immune response.
Okay, all right. Like they bring
cells recognize something, then they present something. B cells recognize that, very simple, and they develop into stuff, and they excrete antibodies to help the immune system and respond to the threat.
Right.
Very, very simple, and I probably got something wrong or not as accurate.
No, that's fine. That's great. That just gives us a basic understanding of it. And when I say us, I mean me, because I'm sure most people know what you're talking about. I'm of course behind on all this stuff. Okay, so then how would you? Okay, but then how would you? So once you found out where the T cells were that there were too many of, how do you tell a system to stop doing it? Is that like a protein or something that you give it that says, hey, enough?
Yeah, or a small molecule like a compound. You can administer those as a temporary relief where you inhibit the response. So let's say signal A gets sent out, but there's too much signal A. You can give people a compound that inhibits signal A, so you only have 50%, and that means there's less T cell development.
Oh,
and I think that's all.
Is it as simple as it sounds?
No.
It
never.
Why is it never as simple? Yeah. The diagrams always make it look very simple.
Yeah, of course, because diagrams are very simplified. I think it already happens with certain people who go into shock or whatever. They get given corticosteroids, and that's like a general inhibitor of your immune response already.
Right, but that's like a sledgehammer when you're looking for something that's just going to
sort of
finesse.
Yeah, but it does help prevent people from going into shock or dying at that specific moment, and then deal with consequences later.
How far away are we from? Is there going to be like will we get over a hurdle and suddenly this stuff will open up? Or is it just a million?
No, I think it just keeps building and building and building.
Okay,
because it's so incredibly complex.
Right. Okay. All right. I was thinking like maybe there was like one thing. I mean, I the cytoflow. Damn it.
Flow cytometry.
That must have been a leap in, because that's allowing you to see stuff almost as it's happening, as opposed to killing it and freezing it and looking at it or whatever we're used to do.
In a way, yes, you do look at live cells compared to compared to dead cells or fixed in like formalin treated.
I'm amazed.
But it both has their benefits.
Why? That's right. I don't know why I suddenly thought of this, but then why were they in England and then sending the stuff to you? Was it because you guys had initiated this project, or because
they had the facilities and the more easy access to the volunteers?
Right. Okay.
And it was a collaboration between our and their group.
All right. So it's not that they don't have this technology. It's just that
no, they could have done it there in house as well, or we could have done it with ours. But it just so happened that this was the easiest solution.
That's great. No, that's got to be cool to be a part of that too. Okay. So Jenn the Nerd said, "Is there a battle for the best and most coveted micropipette in the lab?"
Oh,
there Jenn's questions.
There are certainly discussions on which volume or which type is the best.
Really?
Because we have different ranges. We have ones that go from 0.5 microliter to 3, from 1 to 10, 5 to 20, 20 to 200, and then 100 to 1,000.
Yeah,
and are definitely.
And this is just how much fluid can be.
How much you can accurately pipe it, and but there are definitely. I wouldn't call it wars fought over it, but everyone has their favorite for sure.
Yeah. It's not like the Apple PC wars. There's not a
no. Maybe brand wise, but even
what do you guys use in the office? You guys all of course you're using Outlook, so it's got to be all Microsoft then, I guess. Is that
for the computers? Yes. But most of our bioinformaticians actually use MacBooks.
Well, because it's Unix.
Because it's a bit more flexible with programming, etc. Yeah.
Are you a computer nerd at all? Do you like your computers?
I like building them, but
that's fun, isn't it?
Yeah. It's like a very complex. Well,
not now because it's too expensive now. It's
it's ridiculous. Yeah, I had a scare 2 weeks ago of my GPU dying a little bit. I was like, oh shit, I don't want to upgrade.
Yeah. Because it's
too expensive. Money for it. Yeah.
I've got this little. I don't want to do it because I'll disconnect everything. But I've got a little 2 terabyte SD drive that I just I glue to the back of my laptop. And I picked it up for like $120. $120. It's now $480.
I was going to say that's at least double or triple.
Yeah. And I'm like, I'm worried because it's been around for a while, and I usually like to have a backup one, but I'm like, screw that. I can't. Like, I mean, it's
No, I'm very careful on what to upgrade now.
It's going to be a while. I think it's going to be a while before this stuff comes down to earth and becomes affordable. You know who I blame? AI.
Because that is the main. Yeah. That's one of the reasons why I don't like it either, except for the specialized approaches.
I don't like it that it's killing my building PC stuff. Even the Raspberry Pis are expensive now.
Yep.
Which I also love. Okay. Now I'm geeking out way too much. Okay. Minirockhopper says, Minirockhopper, one of our co-geniuses. Thank you. What pep talk or advice would you give someone who has just made their first big impactful mistake? Sorry, did you get that? I was cackling.
Yeah. In general, or
I don't know. What pep talk or advice would you give someone who just made their first big impactful mistake? I guess, I mean, that could apply. You can apply it to the lab. You can apply it to life.
Well, if it's in the lab, then I would say don't worry about it because everyone destroys something at some point, especially equipment.
Right.
Also, all really? Students, all students blow up like a centrifuge or something at some point. They break something.
Really?
Yeah.
I did it twice.
Wait. There's very much. You can just use it and suddenly the converter decides to poop out on you and it lights it on fire. It happened.
Really?
I once blew up a laminar flow hood while I was working in there.
Wow, how'd you do that? Laminar flow hoods, those things are like this, like the giant kitchen things.
Yeah, but they have a very big fan or electronics setup above them. And I think one of the wires got burned or something. So we had to evacuate the entire floor. That wasn't the best. Yeah, that wasn't the best experience as a fresh student. So
I would say you need a better laminar flow hood is what that's a brand you need to,
Yeah. No, but it was an ancient thing. It was like older than I was at that point. So it was bound to give out at some point.
Right. Okay.
It's just a bummer that it happens, but it always happens around students. So
I keep getting tempted to build my own laminar flow because they're so expensive. And I would love to. I'd like to do some mycelium. I like to do some mushroom stuff. Not the interesting mushrooms, just the tasteful ones. And I'm just sort of fascinated by these sort of alien things. That's why I've got the slime mold going. And I'd love to get a bit, but it seems like a lot of work for a giant fan. All right, Darth Piran. Yes. Now, Darth, you had a question before. Is this the same one? I'm curious to know, because Darth Piran was someone who emailed in advance about. Oh yeah, it's the same question. Okay, good. That's how keen people are to talk to you. Just so you know, they're emailing me before the thing.
That's good to hear, actually.
I know.
I thought at first, like, who's interested in this? But apparently, quite a lot of people.
Well, I am, that's. I'm sure I speak for many people when I say this is. I just find this stuff fascinating. All right, do you know anything about familial Mediterranean fever, a disease I live with other autoinflammatory diseases? If so, do you see a cure for them happening ever?
I have heard of it, but I am not clear on the specifics.
Yeah, I don't know much about it.
Well, it's a familiar
autoinflammatory.
Familial Mediterranean fever. I'm just going to look at on Wiki.
Yeah. What do you use for looking stuff up? Do you just go with Google, or are you like, what's your?
Yeah, a lot of Wikipedia, and then I look up papers afterwards.
You know, I donate to Wikipedia every year because I'm like, it's the most extraordinarily useful tool, and of course, AI is being trained on all of it. So,
it is
okay.
Yeah, it is a genetic disease. So if they detect it quick enough, then maybe. But I think it's very difficult to cure something like that. How do you call it?
But this is the kind of thing you would be trying to cure.
Ideally, it would be. But if it affects the immune system, then potentially, yes, you could fix it. Although not currently with the technologies we have.
Okay. I mean, sorry, and this is sort of I don't even know if this is an answerable question, but I mean, how much of our immune system do we understand at this point? Where are we?
The general bits we do, as in what most cell types do, is what we understand, but like the workings behind that, and especially how it develops, is something of a black box still.
Right.
Of course, we know what we need we know a lot of bits and bobs and pieces, etc., but we don't know how the entire picture goes together yet.
But when you were studying, was that in an area that got you? Like when you were back before you'd specialized, was the immune system something that you found particularly interesting, or just happened?
We didn't get much about it because it was we were mostly focused on like the very basic stuff like cell signaling and cell development. But I did always have a reasonable interest into it. Yeah, I did like the courses that went over it.
It's a fascinating system, and I had no idea how dangerous it was. Like I had no idea how much damage it could do to yourself with your own immune system.
Yeah. But most stuff in your body can kill you just as easily if it wants to. Luckily, it doesn't.
You say that with such glee. Trust me, I can kill you with anything. That's that CSI thing again. All right, Stigma Crier says, "What's the biggest gap between what we could detect in the laboratory and what doctors can actually use on a patient?" Wow,
that's quite a gap. That's a difficult question. We can detect the very great many things, and we can treat very many things, but yeah, what's the biggest gap? That's very tricky.
What's the biggest gap? I mean, what's the biggest gap when you detect? And I guess detecting it's only part of the problem, right? Once you detect it, you still have to figure out how to solve it.
Yeah, but it is a good starting point, of course. Once you detect something, you have something, and then you can work towards it.
They have a colon cancer screening here now, which is I guess is a blood test now, which used to be a lot messier. But the fact that they can again, the easier it is for people to know that they've got stuff, I guess, the faster you can get it into.
Like I said earlier as well, early detection is a very important thing.
So do you see a future when you'll go in, get a blood test, and they'll just test you for all of the stuff? Like
you can never test for all of the stuff, but it is increasing for more and more and more and more and more. You see that with genetic screening of newborns as well, or even like pregnant women can get screened for diseases that the potential, like the embryos might have already.
Yeah, that's we had there was a bunch of screenings that we did when my Bas was born, or before he was born, which I found extraordinary that they could, that, you know,
and those screenings keep expanding and expanding.
It's weird because you get into this almost sort of you know, eugenics kind of thing where we're now we talk about us. You talk about us choosing where our evolution is going. We're literally being able to sort of pick and choose at this point.
Yeah, it's we're very lucky that we can't really see how well someone would develop if it will become a proficient pro athlete swimmer, for instance. We don't have that technology yet.
Do you think it's coming?
Maybe eventually, but that'll be like years and years and years and years, like decades away.
Well, and I feel like we sort of do that anyways, don't we? We do that. We just do that by hunting down people who might have, who have genetic advantages to being a swimmer or whatever. That's what we go and handpick them. Ideally, what I guess, you know, the evil scientists want to do is
we do and did it with food crops, etc. We've been doing it for like ages.
Well, and we're having
sorry,
and we've got problems because of that's the other thing that I found fascinating. This whole monocrop thing. Like, if we create humans that are all the same, are we at risk of having us all wiped out because of it?
Yes, that is a concern, but it's not a concern. We, as long as you have good enough medical care, you might be overcoming that.
Yeah,
I suppose, and that's also part of why we're not like subject to evolution as much anymore.
It's funny how one of the things we were talking about bioinformatics. I went to a bioinformatics conference. I actually spoke at it. I talked about the need to that they needed to get better at how they presented their technologies, because everyone immediately, when they think of bioinformatics and computers and AI and stuff, they immediately think of like Terminator or just these terrible things that they need to start humanizing. It's one of the reasons why I like talking to people, is that you put a human face on the stuff that's happening now. There's not some crazy scientist doing weird stuff. This is someone who's, you know, contributing to the health and improved lives of, you know, of future generations. And the reason why they're into it as well is so important. Whereas when you hear just corporations announcing that, oh, we have this test does this and this, it can feel a bit, you know, sort of commercial and not, you know, connect on a human level. So, okay. Questions. When you fill in for colleagues, what's something you especially like having the opportunity to work on? Ooh, good one, Susan. Good question.
I think I just very much enjoy helping the other people out, getting their research like to keep it going.
Yeah. What's the sort of stuff you get asked to do? Like, what's the
take care of their cells, very basically, because those are easy to transfer, of course, to your colleagues. Everyone does a little bit similar things, so it's easy to take over. But sometimes I have to run essays for other colleagues of which I'm specialized in, and then they want to try something else, and they haven't done it before, then they ask me to do it, or at least show them to. I do a lot of flow cytometry analysis for other colleagues if they're unavailable.
Right. So, any excuse to get on that machine, isn't it?
A little bit, yeah.
Will I see it if I come and visit? Will I see it?
Yes. I'll show you I'll show you the different types. Probably.
Is it expensive? Is it expensive to run?
We have a facility in our hospital, and I think for the very basic machines we pay $15 an hour or something.
Oh, okay.
Euros. Yeah.
Oh, wow. Okay.
But the machines can get very expensive if you want to buy them.
Yeah. No, I don't want to buy them. But I just sort of want to play with it. I don't know why. For some reason, that one really just tickled my imagination. I don't know why. I don't know what it is about it. It's like it's some kind of I remember when laser printers first came out, or inkjets.
Oh yeah.
And I spent hours looking at how they work, because I just couldn't get over these things actually did what they were supposed to do. So
it is. I think the first flow cytometer was the size of a room.
Yeah, and
it could detect 2 colors. Wow. Now we have them like the size of like a meter by a meter by a meter or something.
Yeah,
it can detect. It has 5 layers, and it can detect like 46 things at the same time.
Would you like to run your own lab? Would you like your own lab, like just to have your own?
No. No, I don't think so. No.
You like the team aspect. You
like yeah, like definitely not as a professor or whatever. I don't think I would be a good like manager for that as well.
Yeah, there's too much other stuff that takes you out of the lab on that as well.
Yeah, which we already have with meetings and stuff, administration, making sure all maintenance is done, supplying.
Right. The boring administrative stuff was there.
It's fine as it is.
Okay, and I have to ask you this because we touched on it at the very beginning, and I'm very curious. You went into education for a little while between gigs, so to speak, I guess.
Yes.
And what was the lure there? And then what was the lure back again?
I when I left, it was my previous boss who retired, so the group was kind of falling apart.
Right. Is that what happens? They
retire, and usually there's a like a successor. We didn't arrange that properly, I guess. I don't know exactly why or what happened.
Right. Okay.
I'll not go into details either.
Nope.
So I wanted to try something else, and then there was a vacancy as a teacher in the biomedical. I don't know what biomedical sciences, but on more for work. So it was combined. Like you train people
at Leiden?
there.
Still at Leiden?
Yeah, it was still in Leiden. Like the course or the education trained people to become lab technicians, pretty much.
Oh, okay.
In the biomedical field.
Right.
So, and I was like, oh, I'll try that. And I applied for it, and I actually got the part.
Right.
And then I tried that for half a year, and like all the practicals where you show the people actually working in the lab, I love those.
Yeah.
But the theoretical part behind it was like, it's not my thing, and it gave me too much stress.
Yeah.
And I
you're standing up in front of the students, basically.
Yeah. I missed doing it myself.
Mm-hmm.
And then my previous boss, who was supposedly retired, was still around finishing a PhD project, and he needed a technician for a year. And he heard that I was looking for something else, so I went back there.
Right.
For a year to finish everything, and then I switched to Franck Stahl's group.
Right.
He was very good friends with my previous boss, so
right,
that was easy.
That's great. No, that's amazing. That's so if someone's trying to get into this now, if someone wants to do this, someone wants to become a technician in a lab and do what you're doing, what do they need to do? What do they need? Maybe what are the skills they need? What are the interests they need? And maybe
they definitely need an interest in biology and a little bit of chemistry.
Right. Okay.
That's what you need anyway. And then here we have a specific education, which I taught at for a bit, that trains people to become lab technicians.
Right.
And then you can either choose the more biological side or the more chemistry side, and either is fine.
Right.
Finish that, and then you can apply for a job as a lab technician. It's very simple, but yeah.
How long's the training?
The one I taught at was 4 years.
Wow.
Yeah.
Wow.
Man. The one I did at university was 5 years. It's just 3 years for a bachelor and 2 years for a master. But that prepares you more for a PhD position.
Right. And are you retraining? Do you have to retrain every so often, or you got stuff that, or you do?
No, you keep your experience doing stuff in the lab, and then every now and then people want to try new stuff, and you just learn a new technique,
right,
or manage a new machine, and then you keep growing with that. You can do some courses for data analysis or techniques, etc.
Oh, yeah, I took the Google data analysis course, which was absolutely fascinating. But halfway through, I was using AI to help me with my homework. Not cheating. Ooh. I felt a little weird about it, but I was like, well, you know what it was? It was RStudio. It wanted me to code in RStudio, and I was like, I don't want to code in RStudio. I want to use Python. So I was converting stuff to Python and AI was helping me with some of the code on that. This was before it was good enough to write it all for you. And I all of a sudden they changed the program, and they started saying like, oh, this is how you would use AI to do all this stuff. So it was basically doing exactly what I'd been doing, which was but an interesting course. So do you have to run? Are you
No. I have plenty of time.
Okay, good. I think, well, although that said, I think we're done on questions. I might just pop into the chat and just check if there's anything that came up. Like I normally see on these things, I apologize that I don't get a chance to really interact with the chat, because I do like to focus on our guests and what they're talking about. Otherwise, I find I don't have that. I'm not very good at the split focus on that. That's I'm not a teenager on these things. But if there was, we're talking about these about the the training and getting into this stuff. What about your own? What do you think it is personality wise that allows you to do this and enjoy it and be good at it?
I'd say I'd have to be a bit meticulous. That certainly helps you, because I like to go into stuff prepared.
Right.
You need to like, of course, the subject matter, and you need to like doing some stuff hands-on, not sit behind your desk the entire time.
Right.
You don't have to be very handy, or you can learn everything in the lab.
Right. What do you think about this sort of citizen science stuff, where people are doing this at home, doing their own? I always there's a company that I really enjoy that is trying to create. You remember those electronics kits, those sort of 150 in one? Yeah, they want to do the same thing, but with biology experiments and biology stuff.
Yeah, I think for baseline chemistry experiments, etc., it's very good to get people interested.
Chemistry sets. I remember the chemistry.
Yeah, or even for biology, if you get a little kid, give him a microscope, and then you have all those prepared glass slides.
Yeah, they're fantastic.
Things on there.
I've got stacks of them. Yeah,
it's brilliant.
Oh, I love it. Absolutely,
those are very good initiatives.
Yeah, that's the microscopic stuff is fascinating to me. Like, I just don't. I mean, one of the things we would do on our summer holidays would be we would take a moss sample and throw it under the microscope and try to find tardigrades and things like that. Was which I couldn't believe how many there were. Like, I did those little suckers are amazing to me.
I hope to never find any in my cell samples, but
yeah, that would probably be a bad thing. Yeah. So if you do find any, you'll know where they came from. That'd be me. After the visit, if there's suddenly tardigrades running around,
we'll make sure all your stuff is isolated properly. Don't worry.
That's right. We don't want to get any acting on anybody.
No, they'll start acting up.
Yeah, exactly. Oh,
sorry.
No, it's good. Okay, so some geeky questions. Are you reading sci-fi?
Yes.
What do you like?
Not currently, but because I'm reading the Mistborn series, which is fantasy.
Oh wow, you're like the third person who's mentioned that.
It's very good.
Is it?
Yeah, I think so.
It seems like a long commitment. But it's more fantasy than science fiction.
Yes, absolutely. Yeah. No, but I read the entire series of The Expanse.
Oh yeah,
Foundation,
I've only read the first book.
Yeah, my favorite is it's a very good read. It's 9 books, but it's very worth it.
The Expanse is 9 books.
Yeah, I think it's 9.
Holy crap! I didn't look. The last time I looked, I had 2.
Well, they're very good. I would read all of them.
They're brilliant.
Yeah. What I like the what is it? Hyperion series.
Oh yeah.
Those are really good.
Yeah. Ancillary Justice. Did you do any of that?
Not yet. It's on the list.
That's an interesting one. It's interesting. I mean, it's not biology related, but it's
what else do I have?
It's sort of an interesting concept. The idea that like a ship is one with its crew. They're connected. And what happens when that ship is destroyed, and then the human has to become not the human, the individual has to become an individual, even though it's just an interesting concept.
Yeah, that's why it's on the list.
What else? What else is on there? Old Man's War. Do you want any of that?
Old Man's War. I started it recently. I love that.
I really enjoy that. Is a really page turner. I find it's a little bit bubblegum, but I mean that in the nicest.
But I think that I was going to say that's. Have you read? I think Starter Villain by the same writer.
No.
You'd love that one.
Really? Okay.
Yeah, you'd love that one.
Okay.
It's about a guy who inherits a supervillain business from his estranged uncle.
Shut up! Seriously? Yeah. Oh my god, that's great. Yeah. Oh my god. Okay, that definitely
fantastic.
Sounds like
yeah.
I'm actually trying to get through. Talking about pretentious. I'm trying to get through Quantum right now. The book about Niels Bohr and Einstein discussing, and I keep stopping and going to see YouTube videos because I don't understand what the hell they're talking about. So I'm like, black body.
I was going to say that's probably a very, very tough nut.
It's an audiobook too. It's actually not, because first off, they've written it very well, and it's also about a lot about them, not just the science. But when they get to the science, I almost invariably have to go to YouTube and just look stuff up.
I get that. I get that. That's very difficult science.
Yeah, it's an interesting. For some reason I feel guilty if I'm reading something that's easy.
Why?
Here's a question for you. Are there any sci-fi books or series or movies or anything that's really, you feel like either truly captured the essence of the lab work, or or at least seem legit?
No, not that I can think of currently. Everything is very much Hollywood made.
Right. Right.
Yeah.
Did you see Splice? Did you ever see Splice?
I have.
I don't know. I know that
long ago. Yeah.
He talked to a lot of people about that, and I'm curious to know how. I don't know.
I can't remember the movie.
Yeah, so neither can I. I don't remember the specifics of it. But I just remember that I remember liking the idea that there was a bridge stage between someone doing experiments and then there being a monster.
I remember hating the retrovirus episodes in Stargate Atlantis.
Oh God,
because it's like that's not how it works.
I'm so sorry.
Don't worry,
they're sci-fi nerds. They're not. They're space nerds. Not.
It was still very entertaining, but it was like, oh, that's not how it works.
I'm sure. So, I always joked about the doctors in Stargate because they just killed everybody. I was like, oh God, if they get Beckett shows up, you're a dead man.
Do you mind if I go to the bathroom real quick?
Oh no, of course, no, please.
I'll be back in it.
I'll check.
Sorry.
No, not at all.
You can do your dance and stuff.
I'll yes, please let me entertain you.
I'll be right back. Sorry.
Yeah. You go. No, not at all. Go. Lab work is slow and tedious. Jen, it's true. But that's the fun part. For some people, I think that really, I guess that's what they need. Jurassic book back in the day was pretty decent. Oh, that's just have to ask him about that. That's a good one, Maddie. Thank you. John Scalzi, Old Man War. Yeah, I love that. But I also love his. Oh, he wrote Red Shirts. That's right. I haven't read that either. What else we got? Anyone reading Silverbug stuff on? I'm reading Doctor's Orders. Oh my God, there's all sorts of stuff going on. Wheel of Time, Mistborn. Look at you chatting away. Time to Clone McKay. Yes, and Evil McKay. That's what I've been saying this forever. I'd like an Evil McKay. This is so much fun. God, I just love talking to smart people about doing cool stuff. Again, I'll take this as an opportunity to say thank you to Patreons for making this so much easier. This is wonderful. I just love it. I love it. Hereditary rates apparently straightened up, created the twins, had them planted, blah blah. Where are we at? Just flicking through here. YouTube Premium, yeah, I do that too. I can't stand the ads, just cannot deal with it. What else we got here? We've got, I think that was I refuse to blah 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 blah. YouTube, some people have ads on their channels even when the live. I know, I don't understand. I don't know how anyone uses YouTube without paying, to be honest. I don't. I mean, I know that not everyone is in that position, but I find it unusable without it. There's just too many ads. We're feeling much more relaxed, less stressed.
Yeah. Sorry about that.
No, not at all. God, you kidding?
It's the nerves that are getting to me.
Yeah, I'm sure that and the coffee and the right. No, the tea. It's the tea.
The tea.
I find tea very bad for that. Okay, so no films really seem to have captured it for you. What about books? Are there someone just brought up Michael Crichton, and Jurassic Park and those kind of things.
Bit more, yeah.
Yeah.
Like it's especially it makes sense that they didn't have full DNA samples and they had to splice in some other things.
I love that aspect.
Yeah.
I thought that was really, because I felt like I'd learned something when they, and then they went too far and started having mutant T-Rexes and stuff.
I think I only read the first book. So I love the first movie.
No, it was the movies. It was the movies. Yeah, no. The movies got silly. They started having super smart mutant T-Rex.
Yeah, that was ridiculous.
Yeah, dinosaurs aren't freaking cool enough already. I don't know.
Yeah, I get that. Oh, I don't really know any. I still love the mad scientist depiction in Back to the Future, though.
Yeah,
that's still one of my favorites.
It's funny, one of my favorite science fiction shows now is Rick and Morty, because I think conceptually the stuff that they come up with is extraordinary. Like, have you ever watched it?
Yeah, a little bit in the first season, but I never really got into it.
No, it's not my type of humor. It's too crass for me, but my son was watching it, so I was sort of forced to make it through more episodes than I probably would have sat through. And it got to the point where I was like, holy crap, this, like, conceptually, sure, it's about a guy sitting on a toilet, but the concept that they're selling is so much greater than what than the sort of the tools they use to get you there. I was amazed at that. I honestly, of all the science fiction I've watched recently, that's the one that has surprised me the most. Did you see Project Hail Mary?
No, I haven't had a chance to yet. I do have the book in my cabinet, so I'm gonna plan on reading that. But
well, here's the thing:
I didn't like The Martian as much, so I think that.
Oh, didn't you? Oh, I love The Martian.
Yeah, I think that put me off of watching this one right away.
I like the book and I like the movie, but I like it in different ways. This I was not a huge fan of Project Hail Mary. It didn't feel as quite as sort of tight and sciencey. I like, obviously I like the sciencey stuff, and but he's trying to tell a good story. I get that, but that's the science fiction I love. I love science fiction that I can draw a little, where I feel like I might actually be learning something in the process, which is just kind of fun. Project Hail Mary is, I mean, look, it's a fun, definitely a fun movie. Okay, so what else? Anything else? And then game wise, are there any good sci-fi games or anything? Are you doing
sci-fi games? Oh, let me think. No, I'm playing Baldur's Gate 3 now, which is very much fantasy.
Right.
I play a lot of Dota 2, which I guess you could always also.
Of what? Sorry, of what?
Dota 2.
Oh, what is it? That stand? It's
Defense of the Ancients.
That's okay, right? Yeah,
it's similar to League of Legends, if you know that one.
Yes. Yeah. Oh God, you kidding? I've been playing Riftbound, the card game.
Okay, I haven't picked that up.
It's fun. It's not. I don't think it's quite as. It's not quite as sort of as all encompassing as Magic the Gathering. But I loved. Did you watch Arcane? The series Arcane?
Yeah.
I loved it.
Very good.
Absolutely gorgeous. Those characters came to life. I didn't like the second season as much, but the first season I thought was absolutely brilliant. And my son and I used to play. We used to play League every so often. I'm terrible at it, but my God.
Oh, I'm terrible at Dota too, but friends I play with make up for it. So
yeah. Oh, good. Yeah, that's what you need. You don't have to outrun the bear. You just need to outrun your friend. That's all.
Exactly.
Yeah, that was my friend Cal in the chat here was very good about keeping me alive in when we were playing.
Nice.
What was it? Was it Fortnite? Fortnite was the one we used to play with my son, and my son used to just take great glee in just killing me over and over and over again.
I get that.
Yeah. All right. Well, look, I
play a little bit of Factorio, which is probably up your alley.
Oh, Factorio. Oh, so no, that's too much. I can't.
Yeah.
I don't think I loved Minecraft. Absolutely loved it. I loved the redstone. I loved the fact that you could do so much with it. I was very interested in it from an educational standpoint as well. And so Factorial, I just felt like I couldn't, like I knew I was I'd get lost.
Yeah. It is complex. Let's keep it at that.
It's not a game though, is it? It's more like a building platform.
Yeah, it's a simulator, pretty much.
Simulator, that's I guess a better term for it. Yeah. Are there any good biological? There must be. Someone should do a good, bio simulator game. There was one that used to be able to create life. Used to create different traits to get. Yeah, Spore. That's right. Yeah. But I mean, it had a lot of promise. I'm not sure it went as far as I'd.
No, they promised more than they delivered. So which is a shame.
As always, you've got to market the crap out of it, right? Anything you're looking forward to coming out? Anything that any games or anything that you're excited about?
I'm looking forward to the remake of Ocarina of Time.
Oh yeah,
coming this year somewhere.
Yeah. And so when was that? That was last on what, the Nintendo?
The 64.
64 was it?
I think it got released in 96 or 98 or something.
Wow, that's the time. So now's the time to go and buy one of the old cartridges.
Yeah. I think I still have mine somewhere.
Do you?
Yeah. Not boxed though, so it's not worth anything.
It's not about. But what do you do? Sell it? I'm not. I just think it's kind of fun to have. The biggest issue I find now is you've got to convert everything to these LCD screens, right? It's not. They don't have the old CRT screens that. It's funny. I was very tempted. Then I thought my wife would leave me with an old Commodore 64 that I saw for sale that had the old CRTC screen and the little floppy disk and a disc cassettes and stuff. I love all that stuff. So Welcome to the dark side, indeed. Okay, well, look. Let me just double check, make sure there are no more questions. That I'm not going to leave anybody out here.
People can send them to me on Discord if they think of questions later on. Anyway, so
I was going to say, how do people get in touch with you? How do you,
probably Discord's the easiest.
Yeah. Are you on our Discord now? You're on our. Yeah. I love that people do that. Like, I've got to spend more time on Discord, but there's just always 1,000 things to do. But so basically, if you join our Discord, the Nerdia, as I call it, there's a bunch of people who I've spoken to who are on there now, and we're actually seeing some cross pollination between, the quantum people are talking to the gravity people, and who knows, maybe there's some. Well, I know one of the quantum people wants to put living cells through to entangle them. So there's some.
That would be very tricky, I think. But interesting idea.
Yeah, I thought that. Well, they're basically this is Lisa, our quantum queen, and she's trying to figure out, like, as is many people, where's the line? Where's the line between the quantum realm and the Newtonian world? Like, what how large can things be before they, they're not they're no longer have quantum.
Interesting.
Yeah, isn't that neat?
Yeah.
All right. So are you a LinkedIn person? Do you do LinkedIn much?
I have LinkedIn, yes.
Yeah. I've started using it a bit more for this because I feel like
I saw your invite this morning, I think, or this afternoon.
Oh, did you? Oh, good. Yeah. I just always like to say hi, just just in case there's add you to the list. I never use LinkedIn. I don't know.
No.
I mean, I don't know why.
I don't care for it either as much.
I don't like social media anymore. I've sort of got to a point where did you? See, I did, because in the early naive days of Twitter, when it first came out, I loved it. I enjoyed so much just being able to it was like having a little diary that, for the world.
Yeah. But it doesn't do that anymore.
No,
I find
no. Now I find it stresses me out. I find myself suddenly outraged about something that I didn't even know existed when I first showed up. And I don't like LinkedIn, and I find LinkedIn is becoming more sort of a social media platform than it is a work platform. I just really liked it for organizing the people who I've spoken to. Like, it's just really nice to know, you've got this is their business, the business front, if that makes sense. So
that's fair.
Okay, so they'll get in touch with you. Discord, fantastic. So that's another great reason to join our Discord. Woohoo! Nice marketing there, boss.
You're welcome.
And let me see. Is there anything we haven't talked about that you want to talk about? Is there anything that came up that didn't come up that you wanted to address?
So. No,
yeah.
I don't think so.
And if people want to know more about the lab itself, it's through It's through the University of Leiden, right?
Yeah, at the Leiden University Hospital.
Okay,
is where I work.
All right. And they have internship programs. I mean, could people be applying to intern programs or whatever?
I think if you need an internship, you can always email the professors or the postdocs.
Okay.
Not everyone always has internships available, but in general, you can always contact someone.
And I'll also have to get a link to that course that you're for the technician course or program.
Yeah, I'll send you the entire program.
That's great, and I'll throw them all in the links and get it up on YouTube and stuff. Bas, thank you so much. This is I could talk forever, but we're over 2 hours now, and
we can do another one in a couple of months or something.
Yeah, perfect. When you've got something, well, in a couple of months, in less than a month, I'm gonna be there.
So when are you gonna come over again?
I hope so. I'm coming over beginning of September. I've got a convention in the UK. And then we go to CERN, to the particle accelerator in CERN.
Oh, that's cool.
I know. Yeah. I know how lucky I am there. That's Agnes pulling the strings. And luckily we talked to a couple of people from there. And then I'm going to go to the European Space Agency, and I'm going to be hanging out in Leiden. You're going to be there I'm hoping you're going to be there for
I'll definitely try to be there.
Yeah, if you can, it'd be great. I just think it's really fun because you just get everyone together. We have a few drinks and just get to hang out. And then I think I'm going to come and visit. I think we've got on the list somewhere. There's a I'm going to come and drop a few tardigrades into your project.
That sounds good.
Yeah. Thank you all, everyone.
And thank you for having me.
Oh God, a pleasure indeed. Thank you for spending so much time with me nattering on. I will say once again a huge thanks to all the Patreons, Co-Geniuses, Psycho Liz, Samantha V, Minirockhopper, Ryan, and Jen, who I should have called out as a co-genius while she was asking questions. She had some great questions, and I'm stealing that one about what's on your desk because I think that's brilliant. And again, obviously, Agne, Susan for helping out with everything, and all the wonderful people in the chat. Fantastic. So I will say until we geek again, be brave, be kind, be brilliant. Cheerio, and huzzah, who's actually barking outside as we speak, so I should probably go and rescue her. Thanks so much, Bas. Yeah, cheers.
See ya.